online version came out a year earlier in 2015, where we showed that patients with IBM have patients with who are antibody positive to NT5C1A had more severe motor phenotype with more significantly more proximal weakness, significant reduction in forced vital capacity, much more need for ambulatory devices such as walker and wheelchair, and much higher incidence of dysphagia. And James Lilliker and his group looked at this data in a retrospective study of their cohort from multiple European countries and validated some of our findings, but their additional findings were a reduced life expectancy in patients who were seropositive. Seropositive patients in their retrospective analysis of 300 patients lived about 8 years less than the seronegative patients. Now, more recently, Chris Weil and Chasiko, and you'll be hearing Chasiko later for the work that she's doing in RNA-seq, but she looked at a collection of about 100 plus patients. 65 of them were contributed by us from UC Irvine, and she didn't find any changes in life expectancy. But when she did a sub-analysis of the patients from UC Irvine, there again was this trend of reduced life expectancy, even though it did not meet significance in those cohort of 65 patients within that group, but there seemed to be a signal there again. And then there was a very elegant work by Tawara from Japan, which showed that if you pass on serum from seropositive IBM patients to mouse models, either directly injected into the mouse, or if you put it into a myoblast culture, only seropositive serum samples cause pathological changes, including production of P62 positive vacuoles and inclusions in the muscle. This did not happen with control serum, and they did not happen with serum from seronegative patients. So again, suggesting that pathophysiologically, there may be different mechanisms in IBM based on their serology on that. And then more recently, Steve Greenberg, and this is some of the work that we have recently done. This paper is under review right now in neurology, but this is a very deep phenotyping of the CD8 lymphocytes in patients with IBM. This is a collection of about 51 IBM patients with age-matched controls. And you can see that not only do they have a reverse CD4, CD8 ratio, but they also have a very high KLRG1 percentage within the CD8 lymphocytes. KLRG1 is a marker of highly differentiated immunosenescent T cells. And it looks like that as the disease progresses, there is a progressive increase in these memory cells, especially the very, very differentiated memory cells. So there is an immunophenotypic abnormality that's also going on in this one. So we sort of had thought about a prospective natural history study, and there are multiple current barriers to clinical trials. We don't really understand the disease very well. We don't know if, like ALS, if there are multiple diseases that eventually look the same. We don't know what the rates of diseases are, and if the rates of diseases are uniform in all patients, or they influenced by various biomarkers or other influences. We again wanted to do a more longitudinal study using the biomarkers, whether it's the NT5C1A biomarker or the T cell biomarker, to see if that influences disease behavior and progression. And then obviously these kind of studies require a longitudinal follow-up that cannot be done in a clinical trial. I mean, the Novartis natural history study would have been a great idea, except that the moment the drug failed, the company decided to pull the plug on that one. So to be able to do that, we surveyed the patient population. We sent a survey to about 200 patients, asking them if they would be willing to participate in an observational, non-interventional study. And to our surprise, they were very engaged, very enthusiastic, and they were even willing to undergo muscle biopsies, even despite the fact that this would have been a non-interventional study. So this paper was published in Journal of Clinical Neuromuscular Disorders, and we included an Australian cohort through the help of Professor Mary Lee Needham in Australia. And then we did a community engagement studio, and Rick will recognize this. This is one of the strengths of the NCATS and the CTSA mechanism, where you can get together a group of stakeholders into a room. This was facilitated by one of our former nursing deans. And you ask the questions, what their needs are, what they think is the most urgent need for this disease, and we got some really helpful input into. And this aspect of the trial design, especially the engagement studio and the survey, was a real hit with the grant reviewers. So we proposed a 4-aim study. This was titled as Influence of anti-5C1A antibodies in disease progression and phenotype in IBM. The first aim is to look at a longitudinal version of this question, which is there a different disease progression between the anti-5C1A positive antibodies versus negative in a 2-year period? We are planning to do a detailed histological analysis, because there have been some suggestions that the muscle histology may be different depending on the serology. So we are going to do a subset of patients, about 40, to do muscle biopsy with concurrent blood samples, and then to also characterize further the distribution of the immunosenescent cells, but also look at disease correlation based on these lymphocyte presence. And then one of the questions that I'm interested in is looking at respiratory function. A lot of us have observed that the morbidity and mortality in IBM is associated with respiratory dysfunction, and there is a progressive dysfunction, but it's not been characterized very well. So this is a 2-year study. There are 5 time points for data collection. There is a baseline visit, and the baseline visit actually includes a optional muscle biopsy. There's a 6-month visit, month 12 visit, month 18 and month 24. All of these visits currently are designed to be in person. This was designed before COVID, so we had not put in a virtual element to this on that. And these are some of the procedures we're going to be doing. It's a fairly standard. There's physical exam, there's muscle biopsy, there's handheld dynamometry, hand grip, time get up and go. There are a whole bunch of functional assessment. We are directly comparing CFAT to IBM FRS and see how they perform in that sense. And then we are doing a bunch of other tools, including for dysphagia, for some of the PROMIS tools for pain and sleep disorder. We are using Chat Heat Pulse, IBM Health Inventory, which is a new outcome measure that he's developed. And so we'll be testing it out in the study, and there is going to be a false diary on that. The inclusion criteria is patients about 40 years of age, and we realize that this actually puts it a little bit of a challenge, because the ENMC 2011 starts at 45, so we may have to take some protocol deviation for that. There has to be a diagnosis of IBM. And then for the sake of really keeping it homogeneous and wanted to see the early changes, we decided to restrict patients within 10 years. And there's been a lot of discussion, and actually one of the issues that I want to bring it up in the next steering committee meeting is because we're getting some feedback that this may restrict the number of patients that come into the study. And then obviously they cannot be on any immunomodulation, immunosuppression. Any significant medical history would exclude them on that. We have a fairly wide geographic distribution. Now, we do realize that we are missing a large segment of the Southeast. And we have gotten some requests, especially from Michael Benetar's group, to be included, but I think we'll have to see how the enrollment goes and if we need to add more sites. But that segment of the US is missing at this point on that, but otherwise it's a fairly wide net. These are all the PIs, and some of the PIs are members of the steering committee. There have been some changes in PI. So for instance, Nizar took over from Shafiq, Leo took over from Michael Weiss, and then Salman Bhai took over from Sharon Nations at UT Southwestern. This is the study governance. So I'm the lead PI. We have a physiologist who's an immunologist as a co-investigator, and a very distinguished statistician as part of the team. We have a steering committee that includes Rick as the chair, Chris Weil, Mazen, Rabi as the head of the MSG Coordination Center, which is doing the data coordination as part of the steering committee. Bin Nan is the statistician, and Laura Herbelin is the evaluator trainer. She's going to be going to all the sites to train the evaluators. We have a muscle pathologist, Marie Perez, who's a wonderful young muscle pathologist, but really, really good. And all the immunological studies are done at with the Vialta lab at UC Irvine. And Marie Wenzel, who you know from the Epinema study, is our Uber project coordinator. So the first patient is about to be enrolled. We are going to be enrolling our first patient next week. We are using the Kansas Smart IIB. Thanks, Rick, because that went very well. It's a 6 months of start time, which is, we are about to expire that start time. But the recruitment is expected over the next 9 to 12 months, and the study will take about 24 months from that point. As I mentioned, there's a 40-subject muscle pathology sub-study. So one of the big advantages of this will be there will be a very large repository of patient data, very detailed, well-characterized patient data. We will have PBMCs stored on these patients. DNA is being extracted, sent to Chris Weil's lab. And then we are locally extracting also RNA and keeping it. And then there will be muscle tissue from a highly characterized phenotype group. So the muscle samples will have also concurrent blood samples, so you can actually compare any immunological or any other abnormality. We are locally planning additional studies, including very high-res RNA sequencing. We have now access to mass cytometry and mass imaging cytometry. So we'll be looking at blood as well as muscle samples for other tissues, but that has to be a separate grant. This is not part of this grant at this point. But I just want to draw attention to this very nice, very relevant notice of special interest that just came out. This is from the Aging Institute. This is not from NIAMS, but this is for maximizing and secondary analysis of existing cohorts and data sets, especially in the aging research, in the aging population. And if you look at the NOCI, it actually covers aging related disability and musculoskeletal disorders. So it's very relevant to the IBM population. There are two separate mechanisms. Either you can do it through the parent RO1 or you can do it through the parent R21 for this. So again, we are planning to explore some of these additional opportunities, but there are other sub-studies in plan. I'm working on a sub-study with Leo Wang to look at upper extremity functions as well. So I'm going to stop here. I want to highlight two really exciting developments at UCI. Therefore, they decided to build another hospital on the campus. So this hospital is nicely coming up. It's going to be built in 2024. And then there's a dedicated biomedical research building that's going up that will sort of alleviate some of the space crunch we are experiencing right now. So I'm happy to take any questions if you have time.
Could you go back to your timeline slide? Could just reverse it? Yeah, that one. So how long is this study funded for?
5 years.
And it's
so study. So we gave ourselves a nice cushion of about 18 months for data analysis and cleanup.
That's good, but you're enrolling now?
We're starting, our first patient is scheduled for next week, I believe.
All right. When is the steering committee that I chair going to meet?
We're hoping sometime this month or early November.
All right.
Because I mean, a lot of the sites are not online yet, so they forced some of the.
I see. So we're waiting for that. Okay. Yeah. And then once this is done, how I assume we're going to be able to leverage this group to do clinical trials.
True. So the idea is to create a myositis consortium. These are already experienced sites in IBM. They all have a lot of expertise in it, but we would have trained evaluators, and the idea would be to attract the pharmaceutical industry to pick these sites as a trial-ready site as well. But then there are also opportunities for additional sub-studies, either using the consortium and the mechanism there. So for instance, the PET imaging study that Colin Quinn talked about, that's something that we are planning to do through this mechanism.
So Tony, I'm hearing two things. One, we're out of time. Michael Hanna wants to say something. Tony has a question. Michael, what did you want to say?
Very quickly, that was a nice presentation, important study. I wasn't quite clear. Are you doing 2 muscle biopsies or 1?
Just 1.
Okay, that's fine.
So Amato says it may be tough keeping patients in natural history as promising agents may be coming to trials. Well, Tony knows more than I know.
So we are only expecting 12 subjects from each site. So each site has way more than 12 subjects on that. But as Tony said, there is the sirolimus study, and then there is going to be a immunological treatment with Steve Greenberg's drug, which is going to be coming out in the first quarter of next year. So there may be other drugs coming down the pipeline.
I'm working on one too, but I got to get it funded.
Okay, I haven't heard anything about it, Rick. So, but that's why we kept the expectation from each site to be low. So they will still have plenty of other patients to put in the trial.
Yeah.
Hopefully, I hope it's not become an issue.
All right, very good. Thanks, Tassin. That's great. All right, now I'm turning it over to Tracy. I'm looking at my color code here.
It might be me, Rick.
James, all right.
It might be me, Rick. Yeah, is that okay? Can you hear me?
Yeah.
Yeah. So it looks like we've got next speaker is Mona Hanaini, who I think I just saw on screen a second ago, who's taking us through some real-world registry data on Duchenne. Mona, over to you.
Okay, so good morning, everybody. We are excited today to share with you the respiratory profiles in Duchenne muscular dystrophy patients taken from a real-world data registry. Now, this registry is the Canadian Neuromuscular Registry, and this study was supervised by Dr. Craig Campbell, Aaron St. Laurent, and the statistician Mike Miller and research assistant Matt Downs. I do not have any disclosures to announce for you today. So I will take one moment to highlight on the CNDR, since this study aims at giving a better understanding of neuromuscular diseases through providing real-world data from neuromuscular clinics, and thus helping in facilitating research to develop new therapies and benefiting patients in the first place, their families and caregivers. Now, the CNDR up to date has so far more than 4,500 neuromuscular patients registered across Canada. So what were the main objectives of our study? First, to describe the respiratory profiles from a national DMD real-world data set and to explore the relationship between the FVC and some of the disease parameters such as scoliosis and ambulatory status. So we reviewed 515 DMD patients registered in the CNDR, and most of the patients were from Ontario province, followed by British Columbia and then Alberta. And 101 patients were omitted due to females, different diagnoses such as Becker muscular dystrophy, and incomplete data. So we were left with 414 patients with an age range between 2 and 36 years and average age 14.7 years. Now, 85% of the patients were on steroids, and 325 patients out of the 414 had one pulmonary function test. Now, looking at the parameters, we see that nearly half of the patients, around 51%, were non-ambulatory or walked with support, and 47% walked without support, like they were ambulatory. Now, for the ventilation status, the majority of the patients, around 76%, were on room air, didn't need any kind of ventilation, and the rest, 20%, were distributed between part-time and full-time non-invasive ventilation, and only a minority, around 4 patients, needed invasive ventilation. As for scoliosis, 66% didn't have any scoliosis, and 28.7% had scoliosis, of which 28% were operated. Now, back to our results. We say now the like the latest visit was taken for the patients with the full data information and recorded FVC, and we see here in this first scattered plot that two variables are being compared: the FVC and the age. And upon regression, upon applying the regression model, there was a statistical significance between the decline of FVC and the increase in age, in terms that with every 1 year increase in age, there was an average decrease of FVC around 3.19%, which is pretty similar to previous natural history studies done in 2015 and 2017 that reported a decrease in FVC between 4 and 5% with every 1 year increase in age. And upon applying the survival analysis, patients on average reached the FVC less than 50% at around age of 21 years. Now, looking at the second scattered plot, there wasn't really any statistical significance in the decline of FVC between the three groups of scoliosis. The black dots are the patients who didn't have scoliosis. The red dots are patients who had scoliosis and wasn't surgically corrected, and the green dots are patients who had scoliosis and were surgically corrected. So running the ANCOVA, there wasn't any statistical significance, though visually, when we look at the graph, we can see that patients with the scoliosis and weren't surgically corrected had a rapid decline. And again, the survival analysis didn't show any significant difference between the scoliosis groups in terms of age at reaching FVC less than 50%. On the contrary, when we compared the ambulatory groups, we found statistical significance for the decline of FVC between the ambulatory groups and the non-ambulatory groups, in terms that with every 1 year increase in age, the FVC declined 0.96% in the ambulatory groups, the green dots, and 2.47% decrease in FVC in the non-ambulatory group. And upon running the survival analysis, there wasn't any significant difference between ambulatory groups in terms of age at reaching the FVC less than 50%. Now, this last graph is a bar graph that shows us some description for the ventilation status of the patients divided into age groups. So we see that patients below 12 years of age didn't need any kind of ventilatory support, the blue color, and starting from the age of 12 years, they started needing ventilatory support, mainly the part-time non-invasive ventilation, and between 16 and 18 years, they started requiring full-time non-invasive ventilation, and as they grew older, more patients were on ventilation rather than on room air. And we see also that the majority of the patients were less than 20 years of age. So few conclusions to say here. First, again, highlighting on the Canadian Neuromuscular Disease Registry, which is a Canada-wide disease neuromuscular registry that provides real-world data. And it's really a valuable tool to be used for research and to facilitate research and collaboration studies. And it reflects the information taken in the neuromuscular clinics. Now, the profile for forced vital capacity and ventilation status was similar to other natural history DMD studies. And as anticipated, the forced vital capacity decline was more significant in the non-ambulatory group. However, in the group that had evolving scoliosis, there wasn't any statistical significance in the decline of FVC. And this may be due to that this is a cross-sectional study, and maybe when looking at the longitudinal study, we will find the statistical significance between the decline of FVC and scoliosis. So from this point going forward, we will be looking at all of these factors in a longitudinal manner, and we will be specifically looking at the ventilatory support and its impact on FVC, and also the role of steroids and their impact on the FVC too. And we will be presenting further the results of this study in pulmonary in future pulmonary conferences. Thank you.
Thanks very much, Mona. Really interesting talk. We do have a couple of questions. We've got Michael Pulley. I'll just ask him to unmute. Michael, are you there now?
Yeah, I just wanted to, in the category about ambulatory status, it seemed to be missing walking with assistance. There's only unassisted or non-ambulatory in the early slides.
Yeah, exactly. I said it's written non-ambulatory, but when I was explaining, I said non-ambulatory or walking with support. And the other group is ambulatory, like they walk without support, without any part-time wheelchair or anything.
Does that answer your question, Mike? Oh, sorry. Mike, do you want to come back in on that?
It didn't seem. I was the first category that there was like 100% of patients accounted for, but nobody who walked with assistance. So I didn't quite understand like the proportion of patients who were walking with assistance. It was only non-ambulatory or ambulatory without assistance, and so there was a group of patients that were missing or not captured or something. I don't know.
If I got your question correctly, so the 414 patients were divided between unknown, we didn't know their ambulatory status, and fully ambulatory 47%, 47.58%, and non-ambulatory or ambulatory with support 51.44%.
We got the slide there, and perhaps whilst we're reading over that, Michael McDermott, do you want to come in with your question?
Sure. Nice talk, Mona. I had a question about the survival analyses you did. It looks like this is cross-sectional, so that you recorded the age when they had their last FVC recorded. But it sounded like you were reporting the age at which they first had an FVC that went below 50%, but I'm not sure that that's what you have. Maybe I'm misunderstanding something.
Okay, so for the regression model, we used the FVC, the last FVC recorded with the age at this last FVC. Okay, now for the survival analysis, it was used like the age, also again the age at this last FVC. And like we had the help of our statistician. Like I know that this here it's a little bit like 21 years. Like usually what we know from like looking back into previous studies, it's more like around 17 or 18 years. But again, this is a cross section, and maybe in the longitudinal study we would like running the survival analysis. It would be like a bit different results.
Yeah, I would suspect that the time when they first reached 50%, less than 50% FVC is younger than what you actually have there. That's right. Yeah. Okay, thank you.
Good stuff, and thanks very much, Mona. Working with this real-world registry data does create lots of its own challenges, and I congratulate you there on that analysis and a very clear presentation. So thank you very much. I think we're going to move on now to the next session, which is the Shark Tank session. So I'll ask our three other sharks just to share their video and unmute themselves whilst I just do a few introductory remarks. So this session, as Rick was saying, I think it was Rick's idea last year for the first session, and the idea is to sort of a version of the Shark Tank TV program, which we don't have that in the UK. We call it the Dragon's Den, but I think it's very similar, where some young entrepreneurs present their idea in a couple of minutes, and then they're really quizzed about the details in a sort of interactive session afterwards, and really they're fighting for investment in their company. So,
unfortunately, James, I think it was Trump's idea, but let's not go there.
Oh heck, right.
Let's just.
Well, it's on the BBC in the UK, so I'm sure he'd love that. So what we got here is some young investigators. They're all, if you like, starting out in research. They're going to present an idea for a study that they're working on. They're going to present in, as I say, a short period of time, about 5 minutes or so. But there's going to be about 20 minutes or so of questions, and there's going to be questions from my fellow sharks, who shall introduce themselves in a second. And then also I'd invite questions from the audience. So please get typing in the chat. Make yourself known if you want me to unmute you. I'm more than happy to open up the discussion, and we'll draw each presentation to a close after 20 minutes of questions. We've got three really great pictures coming up, and with no further ado, then I'll ask the sharks to introduce themselves. I think I can see David on the screen in front of me. David, do you want to just introduce yourself briefly?
Sure, it's a pleasure to be here. Really happy to take part in this, and I guess if I could choose between a shark and a dragon, I think I'd rather be a dragon.
Yes, it's the breathing out fire.
That's right, exactly. But I'm a friendly dragon though, so.
Okay, and Geeta.
Oh yeah, hi. Well, hopefully some of you saw me present yesterday, so know a bit about me. But I work in London at Queen Square. Just to correct the slide, I'm not DPT. I am a physiotherapist. I'm a consultant therapist here, but I'm a PhD. So hopefully that will be helpful.
And Tracy.
Yeah, hi, I'm Tracy Willis. I'm a paediatric neurology consultant in the UK, working in Birmingham and Shropshire, for anybody that knows that. Yeah, I'm just really excited to sort of see the presentations today. And yeah, I think I prefer myself being more of a dragon rather than a shark.
And I guess I should introduce myself as well. I'm James Lowick. I'm a neurologist working in Manchester, which is quite close to Tracy, actually. So we've got a good, some good waiting here from the north of England, which I think is particularly important. Yeah. And on that point, actually, our first presenter, Catherine Dodd, is from the same institution as me. So here, full disclosure, I'll probably take part in the questions and things, but I won't be voting on Katie's presentation. I'll recuse myself. So, Katie, are you there?
Yeah. Hi.
Hi, Katie. Do you want to share your screen?
I'll just share my screen.
I'll be timing 5 minutes, and I'll be sort of relatively harsh with cutting you off, if that's okay.
Yeah, that's fine.
But we'll try and keep it fair amongst the three of you. So whenever you're ready.
Okay, lovely. Right. Thank you very much for the invitation to present my research proposal. I'm Dr. Katie Dodd. I'm a neurology clinical research fellow and PhD student in Manchester. And my project's entitled Circulating Myeloid Cells in Myasthenia Gravis. I'm being supervised by Dr. John Sussman, clinical neurologist and author of the British Guidelines for Myasthenia, and Dr. Madhvi Menon, Presidential Research Fellow in Immunology and Inflammation at the University of Manchester. So myasthenia, we know, is a prototypical autoimmune disease, but there's great heterogeneity within the condition. There's a wide variation in severity and response to treatment between individuals, and it's difficult to predict treatment requirements at onset, given the poor correlation to antibody titer. other aspects of an individual's immune system, presumably therefore impact upon how a disease manifests. We know that different pathophysiological mechanisms initiate the disease, for example, paraneoplastic antibody production from thymoma, thymic hyperplasia in early onset myasthenia, and thymic atrophy with loss of central immune tolerance in late onset. Ultimately, these processes all lead to failure to eliminate autoreactive T cells, which then stimulate B cells to produce the pathogenic autoantibodies. Although autoimmune diseases are seen as abnormalities of the adaptive immune system, increasingly components of the innate immune system are recognized to play a key role. However, changes in this important cell population in myasthenia is understudied. Myeloid cells act as antigen-presenting cells, for example in the thymus, which prime and activate autoreactive T cells. And aberrant mature dendritic cell populations expressing cell surface adhesion receptors have been seen in the thymuses of those with early onset myasthenia and thought to result in the prolonged presentation of self-antigen. At the target tissue of autoreactive T cells, the muscle end plate in myasthenia, components of the innate immune system recruit T and B cells and enhance their effect, which can become self-perpetuating. Muscle end plate inflammation and necrosis in myasthenia shows macrophage infiltration. And in experimental autoimmune myasthenia, this can be attenuated by the induction of immature dendritic cells or the adoptive transfer of myeloid-derived suppressor cells. But are there changes that could be measured in the circulating myeloid population in myasthenia that reflect disease activity? Well, it's known that the activation status of components of the innate immune system impact upon the peripheral tolerance and ignorance mechanisms of the adaptive immune system. Myeloid cells are the primary producer of B-cell activating factor BAFF, and the serum levels of BAFF are elevated in myasthenia compared to healthy controls and correlate to antibody titer, although not disease severity. Certain changes in myeloid cell populations in myasthenia have previously been described. A 2019 study of patients in China found significantly reduced circulating plasmacytoid dendritic cells compared to myeloid dendritic cells in naive myasthenia gravis patients compared with healthy controls, which normalized after immunosuppressive treatment. And a study of 40 patients with myasthenia on varying amounts of immunosuppression found an increased prevalence of CD80 positive monocytes. CD80 is a co-stimulatory surface marker that binds to CD28 and CTLA4 on T cells, causing T cell activation. And a 2008 study found that CD14 monocytes express significantly more PD-L1 in patients with myasthenia than healthy controls, particularly after thymectomy and in males. PD-L1 is an inhibitory receptor that mediates negative regulation in the periphery, and we know that inhibition of PD-L1 can cause myasthenia. One of the problems of interpreting studies of immune profiles in myasthenia is the heterogeneity of disease. So validating these findings in a study that accounts for the immunosuppressive status, disease severity, and disease subtype, whilst also exploring other myeloid cell types, would further add to the understanding of these pathophysiological mechanisms and may lead us towards a better biomarker of disease activity. So I propose performing a 15 antibody myeloid cell profile via flow cytometry in patients with myasthenia in a structured way that aims to reduce heterogeneity. This will run alongside my wider PhD project, and so I can compare these measures to standardized outcome measures, measures of immune function and immunosuppression used in routine clinical practice, along with a B and a T cell profile done at the university by myself. And I'm collaborating with Paul Morgan at Cardiff, who will be running a complement profile on these patients. I estimate that to run this panel on 40 patients will cost around $7,000. Thanks very much for taking the time to listen to me, and I look forward to your questions.
Yeah, thanks, Katie. That was exactly 5 minutes. So brilliant timing. Excellent. Yeah, I should say, just in case I didn't make it clear at the beginning, that the prize is $10,000 US. So it's quite a bit to be getting on with. Let's work around the sharks now and take some questions. David, do you want to go first?
Yeah. So great presentation. I think it's a very important area to understand, how to stratify patients. So I guess the big two questions I have is, if you don't get this funding, what's the plan? Are you going to be able to do this? Do you have plans? And then if you do get the funding, what's the next step that you would use this data for moving forward?
Yeah, so if I don't get the funding, if I didn't get this funding, then I do have my eye on another pot of money that's a little bit less. So I could do it on a smaller population of my cohort to start off with, and see if I can find some interesting findings, and then apply for further funds from there. If I did get this, and I think this would allow me to look at it in a more substantial amount of patients, then there's other ways to look at it. So one would be my cohorts at the moment don't have anyone that's just on high-dose steroids alone. So it would be good to look at that population to see the impact that steroids has on it, or to look in a more prospective way. So the way I've set mine up is because I've got to get it done in a 3-year PhD. And so it's a sort of cohort study, and it includes refractory patients that you wouldn't be able to get in as a prospective study, because you don't know, it takes many years to sort of develop that refractory status. So I think getting that initial data now and then validating it in a more prospective study in naive patients, and then as they go into steroids, and then as they go into other immunosuppressants, would be useful.
So are you telling me you don't need the whole $10,000? Just kidding. No, just kidding. Yeah, no, that's great. I'll let other people ask questions.
Okay.
Ita, do you want to go next?
Yes. So with the numbers that you have in the cohorts that you're going to be looking at, I'll put my hand up. I don't know the methods that you're looking at, but how sensitive are they when you're looking at actually small numbers initially to be able to start to get an initial idea of any effect sizes?
I suppose it depends on the changes you're looking for, but as I mentioned earlier, some of these other studies had looked at populations of about 40 patients. As you can see, I've split it down into quite small subgroups, and I suppose it depends on whether there's big enough differences between those smaller subgroups, doesn't it? And I think because it's quite understudied area, it's difficult to be able to do really detailed estimates of sample size based on what the literature is available at the moment.
So with that in mind, have you had to think about your analysis in terms of how you will look at that? How will you make the comparisons between the groups with a view to the future work that you want to do? What might help you?
Yeah, so I mean, what I would like to do is compare between the different groups of treatment requirements. So there's a group not on any immunosuppression, a group immunosuppressed and stable, and then a group immunosuppressed but refractory to treatment. Certainly it would be good to compare between those groups, but also within each of those there's early onset and late onset. So there's ways of sort of grouping, sort of making the groups bigger by looking at it in different ways.
Yeah, just being cautious about the variability, but it'd be very interesting to look at the effect sizes. Great, thank you.
No, and on that point, Katie, do you have a sort of contingency plan if it looks like the study's underpowered?
So absolutely. So I'm recruiting more patients than I've put into this myeloid cell profile, and the reason I'm not doing or planning to do the myeloid cell profile on all of the patients in my wider study is that it has to be done fresh, so it's just a lot more difficult logistically to get it done on everyone, because I have to go and process them the same day. But if I do think I need more, then I am getting more samples from other patients that I'm doing the T and the B cell profiles on, and so I could choose to do further, find a way to process more of the myeloid cell panels.
Good stuff. And we'll bring in our final shot now, Tracy.
Yes, I was just going to ask, obviously I'm coming from a paediatric point of view, but I was going to ask about sort of how are you controlling for your groups, as in their severity? I know you've sort of broadly sort of banded them, but how are you controlling for their severity and also the longevity of the disease that they may have had, and plus or minus thymectomy and no thymectomies as well, as we see that quite a lot with our teenagers that we.
Yeah, and that, I mean, it's the difficulty of myasthenia, isn't it? Is there's so many different heterogeneous groups, and so I've tried to reduce heterogeneity as much as possible by only looking at acetylcholine receptor antibody positive patients, no one with a previous thymoma. I haven't grouped by whether they've previously had a thymectomy or not in the past, but I have grouped it into early onset and late onset, and I suppose it's a sort of post hoc analysis that I would definitely want to have a look at from a thymectomy point of view. Disease severity. So I think that my groups are more based around the treatment requirements of the individuals. So these are patients who have been treated for some time. So they've certainly had the disease for at least 3 or 4 years. And have been the first 2 groups have been stable for over 2 years on their treatment, whether that's immunosuppression or not immunosuppression. And refractory may have had it for slightly longer, but they're not new naive patients, any of the groups. And then I will be recording on sort of outcome scales, the disease severity scale, and certainly can have a look at that as well, and compare that and see if that makes any difference.
And which scale are you using? Sorry.
So the MG composite score.
Yeah. Thank you.
Thanks very much. And Katie, we've got a question from the audience around your definition of what constitutes refractory myasthenia. This is sort of, David comments that this is generally variably interpreted. So what criteria are you using?
Yes, as you say, there's no sort of agreed definition in the literature. In the UK, though, we've set up a NHS England network of refractory myasthenia clinics. So that's 9 centres within England that are allowed to give out rituximab for what's called refractory myasthenia, and that sets out some a definition of who would class for that. Now, I'm not going to go through that because it's quite long, and it includes patients who've not responded to 3 standard immunosuppressants, or patients who are very acute and not responding to sort of normal acute treatments. And so anyone that falls into the NHS England criteria for rituximab will be included in the refractory cohort.
So it's a pretty clear answer there. And we've got Vino coming in. May have missed this. Will you be excluding those who've had recent Plex or intravenous immunoglobulin?
Yes, because the 2 stable cohorts, so non-immunosuppressed and immunosuppressed, should have been stable for 2 years, so they definitely shouldn't have had any. Within the refractory cohort, no, I wouldn't exclude them because they'd had that recently. But I think to be getting the rituximab, I don't think you can give that on the NHS England criteria for at least 4 weeks since the most recent rescue treatment.
Good stuff. And we've got a few more minutes for questions if others want to come in, including the other sharks. But in the meantime, I'll say, Katie, let's assume everything goes to plan and you win the money, your project is a success. What does it all mean? Where will it get us, and what do you plan to do next?
So, I mean, I'm really hoping that I can find something translational into a biomarker of disease severity or of effective immunosuppression, or to predict what the next best treatment would be. Because I think in myasthenia, what's going to be coming out in the next 5 years is newer therapies like complement inhibitors and FCNR inhibitors, and being able to work out or see differences between different patients and on a sort of immune profile level, and being able to direct the treatment would be good. So what I'd like to do is have a really good look at the B cell profile, T cell profile and myeloid cell profile, look at the differences between these different groups, and then hopefully go on to do a prospective study that validates that and working towards translating it into clinical practice.
Good stuff. Geeta?
Just a question about, because if the biomarker that you're trying to relate to disease presentation, disease severity with the outcome measures you mentioned before, So with the composite score, for example, do you know what the minimal clinically important difference is for that? Because I think sometimes what can happen with physiological biomarkers is that you may see change, but actually it's not necessarily represented in the patient's experience. So I just wondered how much, obviously your focus is on this sort of very elegant work that's quite lab-based, but have you explored, actually not just making assumptions about the clinical outcome measures, but actually how they perform? Because it might be you might miss something if you're not really clear on that. Have you looked at that?
So the MG composite score, as I understand it, is validated in clinical practice, and a 3-point difference is the sort of validated significant difference.
Yeah. Just as a thought, if you ever did expand this, and I know I'm coming in from a patient experience angle, but actually whether or not there's an opportunity to sort of understand, because sometimes these scores don't necessarily, by the fact that they're categorized, sometimes the real nuts and bolts of what people experience can be a little bit lost. But actually, I'd be very interested to know in actually what people told you, because sometimes that can be very revealing. And I think that's another study, but it was just to sort of bear that in mind, is that sometimes you lose the granularity sometimes with the categorical scores, whether or not there's an opportunity for that, maybe at a later stage for you to sort of understand to understand their experience of what may be different.
Yeah, and I'm also doing a quality of life score as well.
Yeah, but although that has the same problems.
Yeah.
What would that involve, Geet, just out of interest? Sort of interviews, structured interviews, and some qualitative work?
Yeah, it can be really interesting, actually, because there's certainly, we've done it when looking at how people felt after an exercise intervention, and actually what wasn't necessarily always picked up by the fatigue score, for example. Actually, what people then described did show that they felt a difference. It's just that this to do with sensitivity, categorical scales, as I was talking about yesterday, sensitivity can be an issue. And I think also just involving patients, even at these early stages, in terms of how they think and how they experience is a good shout, as we've just heard from the IBM studies earlier, how important involvement is for, I know that was a natural history study, but this it can be very helpful as well to sort of try and direct it.
Absolutely. Tracy, David, any final comments, questions?
No, I mean, generally, it looks, I think, yes, we need to get sort of more sort of sensitive biomarkers, particularly sort of for monitoring our patients and improving, sort of patient outcome and treatments, and whether what we're giving them is actually making a difference. But I think, as Geeta said, I think, sometimes some of these scales don't always pick up and detect actual sort of improvements that the patients are seeing. Our scales are not very often sensitive enough for that, and qualitative work is very important with regards to that. So I think, going hand in hand, and if you've got 3,000 left over from the 7,000, then maybe you could plug that into, sort of looking at that. So, yeah.
So, yeah, it's not just the scales, but the definition of the subtype. So what is your definition of early and late onset in the British scheme?
Well, it's another kind of worms, really, that there isn't a strict definition, and so different authors will the sort of grey area between age 40 and 50, and I think there's going to be an overlap, and the only way you could do it is something like HLA typing or looking at additional antibodies. What I'm planning to do is just do a cutoff of age 45, and then do some post-hoc analysis, sort of excluding those between 40 and 50, and see if it makes a difference.
So over age 50 is old. Okay, that's the message.
No comments.
Not old.
And then what about other, we have all these great scales, which we've all developed over time. We've got feedback on that, but subtypes of how they present. I mean, do you think whether or not someone has a predominantly ocular pharyngeal or limb, or the interesting distal presentations, do you think that would make any difference at all in the lab biomarkers?
I don't think so, because my understanding is that the majority of patients present initially with ocular because the ocular muscles are more susceptible to disease. And so I presume those who present with, say, bulbar onset, it's the muscles that are different rather than the circulating immune cells. I don't know, but that's...
I mean, that's the big biology question. Why does it? Well, you're right. Most present with eye initially, then it quickly settles out to being either predominantly ocular bulbar or more limb. It's fascinating that some people have the ocular bulbar and have very little limb involvement.
Yeah. But I think the extraocular muscles, the muscle end plate is less folded, is my understanding, and so
there's some suggestions on why. So, but that's a real basic science biology question. I don't know how important it is, but
David, just coming to you last, if you have any other last questions or comments for Katie now.
No, great presentation. No other questions.
Did you want to come back, Katie, on this issue of the qualitative studies and whether you can address that in your study, just to finish that off?
Yeah, no, absolutely. I think it's a really good idea. And for the majority of the patients, I am recruiting from a number of different sites, but the majority of these will be from Salford, which is where I work. And so I'm the direct sort of clinical care person for them. So I think it's a good idea to try and get some quantitative information from them.
You can do it over Zoom quite easily now.
That's it, yes. Yeah. Brilliant. Well, thank you very much, Katie. Well done. And yes, kudos for going first, because I know that's always slightly nerve-wracking. Great presentation, very clear. And we'll wrap up there and move on to the next speaker. So just a quick reminder to the Sharks, perhaps just whilst I'm introducing that speaker, to go to their scoring sheet and just enter your scores and some structured feedback. And just to highlight, all of our speakers will receive some structured feedback on their pictures, which will hopefully help with any future presentations. So do let me know if you can't access that form, guys, or there's any problems. But without further ado, I shall introduce Jenna Lynn Lammers from University of Florida, who's talking to us about the therapeutic play gym pilot study. So Jenna, are you there? Oh, there you are, great. And your slides?
Can you see my PowerPoint?
Not yet, but it's doing.
Oh, great. Oh, there we go. Thank you.
So Jenna, take it away whenever you're ready.
Good morning, Sharks. My name is Jenna Lammers. I'm a pediatric physical therapist and neonatal therapist, and I work as a clinical evaluator for rare disease trials at the University of Florida. There's a problem that spirals out of control in many of our youngest neuromuscular clinic patients, and that problem is that disuse atrophy at the level of the individual muscle cell quickly causes an expensive spiral of secondary medical impairments. These body system impairments then lead to significant functional delays in gross motor, fine motor, language, and cognitive skills. Sadly, we see this problem in both treated and untreated children in our clinics. I want you to meet Brandon. He is a 6-year-old with a rare leukodystrophy. His body system impairments are torticollis, scoliosis, right hip dysplasia, joint contractures, fractures, extremely high muscle tone and spasticity, and he cannot improve his fitness unless he's in an aquatic environment. His parents are concerned. He spends his day in static seating and standing equipment, and when he is on the floor, they have to physically lift his limbs in order to help him access toys or educational activities. I was curious if the play gym I'd been working on for low-tone children with spinal muscular atrophy would work on a high-tone child. So I brought Brandon into our lab, and here he is. He's not able to train in prone at his home, and yet very quickly, once we placed him at the play gym, you can see him training. When he's on his back, he often goes into extensor posturing and his head goes to the right. So we slowly began to correct that by using a very simple cuff and strap system. And we brought his chin into a tucked position so he could have downward gaze and actually see his hands. He's not able to lift his hands on his own to play with the toy. And he started playing with some toys. We then started working on his lower body to break his muscle tone, and it worked very nicely. The one thing, though, is he was not able to keep his foot on the ball, probably because of his right hip dysplasia. He doesn't have as much control on that side. So we did some activities to bring some body awareness to his right hip. And as you can see, by the end of the session, he's not only keeping his right foot on the ball, he's starting to participate with me in some mid-range control activities. For a child with high tone, this is just extraordinary that he could do that first time through. His parents were so excited and asked us, would we consider adding an older cohort to our study? Our study right now runs age birth to 5, and he's 6. And so we're definitely considering doing that. I want you to meet Sweet Pea. She's a 1-year-old with spinal muscular atrophy, and she was born several days before newborn screen went live in our state. We met Sweet Pea at our clinic when she was 3 months of age. We were able to quickly help her get on 2 different FDA-approved medicines. And we are so excited. She is not vent dependent, and she does not use a G-tube. But as you can see, like Brandon, she has significant body system impairments and functional delays. It's because of Brandon and Sweet Pea and other children like them at our neuromuscular clinic. And these children have diagnoses of X-linked myotubular myopathy, infantile onset Pompe disease, Niemann-Rod muscular dystrophy. Been collaborating for the past year with their families to help finish the design for the play gym and then write the pilot study. I want to show you Sweet Pea training here in the play gym. She's getting some nice activation of her left arm. She's getting good weight bearing on the right side to develop her shoulder girdle muscles. She has kyphosis and developing scoliosis, and she's activating her upper thoracic extensors. You can't hear it, but her mom just turned on the music, Baby Shark. And you can see that she's trying to dance. Her right leg cannot dance on its own, but her left leg, with just a little bit of help to unweight her body and fight the effects of gravity, can allow her to dance. She's moving her legs even though she can't see them. We're very excited to be able to offer this pilot study to the youngest and weakest children in our neuromuscular clinic. Our IRB is having us enroll both the child and the caregiver as subject IADs because the families will be doing the treatment at home. This is going to be a 6-month trial with a 2-year extension. And we can only learn so much about safety and efficacy from such small numbers. But what we do hope to learn is, do we have a good study design, and did we pick the right outcome measures? So to evaluate this, we're using the International Classification of Functioning and the F words. The top part of the framework is ensuring that we picked the right quantitative categories to think in. And the bottom part of the framework is to ensure that we have qualitative data as well. If awarded pilot funding, we'd spend $7,000 to make 20 study kits, and then $3,000 to address travel-related barriers that many of our clinic families have. We believe that the future is now to trial how a simple, affordable, portable exercise system may impact outcomes for the most severely involved children at our neuromuscular clinic, and we hope to be able to offer it to your clinics in the future. Will you help me?
Jenna, thank you very much. I'm going to have to come to Tracy first on this one.
Yes, yeah, thanks, Jennifer, a fantastic talk, and certainly it's something that we, sort of need to be looking at, particularly with these more severe children, and preventing that secondary sort of muscle loss. So could I just ask, sort of obviously severe neuromuscular conditions in, sort of infancy covers a huge range. So could I ask you, are you targeting specific conditions, and what are your age ranges and the age at which you want to start this intervention?
Great. In our pilot study, we're defining severe neuromuscular weakness as the inability for a child to lift their limbs or heads against gravity in 3 play positions: supine, sideline, and prone, because those are so important for developing the musculoskeletal system. And so we are not targeting certain diagnoses. We have a number of diagnoses in our clinic, from SMA type 0, type 1, infantile onset Pompe disease, X-linked myotubular myopathy, Niemann-Rod. I mean, those are our most severe. children. I do go around to local clinics. I work with 8 other PTs and OTs that let me join in their treatment sessions. So they do have some other children who've had like traumatic brain injuries, Rett syndrome, that I've been trialing the device on to see how it works. I originally had in my mind that it would really only work for a child with low muscle tone, and I'm learning that's not true. And I'm excited because some of the kids that have the higher, like Brandon with the higher muscle tone from a degenerative brain disease, it's a very simple portable play gym that is working nicely.
Jenna, this is completely outside of my area of expertise, but just for the avoidance of any doubt, could you explain what's different about this than what standard of care currently is?
Yeah, part of this came out when COVID hit. Our children at clinic lost everything. They lost access to their home therapies. They lost access to a lot of their outpatient. Most therapists in our area couldn't even provide televisits to give consultative services. And so what we started to see and listen to is families calling us and coming into clinic and saying they're getting these horrible deformities. And we know they're coming, but it's happening so fast. And so I actually made this play gym. I called the owner of Bennett Corporation, and he helped me with the straps and cuffs. And we were going to just give these to our clinic families. And then we couldn't because of liability. And so the lawyers suggested we do a pilot study. And it really just kind of took on a life of its own. Families now call and ask to be part of the demo. So we've now demoed 18 different families in it, and we're learning from them. The design changes and has changed a lot just because of their input. And it's really fun. The last little girl I showed who was bent dependent, she has very severe scoliosis, and she doesn't really talk when she comes to our clinic because she's so anxious. And yet we got her playing kind of an imaginary game in it. And she was actually able to tell me, I had 2 different straps. One was rigid and one had more of an elastic component. And she was actually able to tell me she liked the one that was more rigid. And I noticed it was on her left hip that was dislocated. And I think it gave her a little more stability. She could initiate more force. It was easier for her to move if I gave her that rigid strap. And so what I love the framework we're using. I love the collaboration with the families. And I love it that the kids are actually giving their input into the design. So,
so one of the key things, apart from the equipment itself, is the fact it can be done in the patient's home. Is that one of the
yes, we're
key
asking
elements here?
Caregivers to train a minimum of 3 times a week, but to record how many minutes they're doing it in each of the positions. And then they are allowed to train more. I just am going to do some extensive training at baseline to ensure that they see the child's fatigue cues and that they stop training when they see those cues. So we don't know how many times a week the families will actually train. Some may just train free. That's the minimum. Some may train more. And we kind of find that when I go into the clinic and try this on the children, every child has a little different training threshold. And some get very angry when you take them out because they can move in it.
Got you. Geeta?
Yeah, lots of questions actually, Jenna. So you said that you're obviously at this initial exploratory. This is an exploratory stage, isn't it, that you're looking at?
Yes.
And so safety is one of the things you're going to look at, and you through sort of adverse incidents and that's fine. And sorry, that the other aim. So did you say it was a feasibility or efficacy? What was the second thing that you wanted to explore with this study if you were to get the money? Safety.
Yes, and I'm using the neuromuscular grow from the Nationwide lab. They've done.
Sorry, no, not even at outcome measure level. What? So is the aim to explore the feasibility of using this or the efficacy of using it?
I think it's hard to establish efficacy on such a small number, and so it is a feasibility study, and yet it's a challenge because there is such a small number of these children.
No, but that's fine for feasibility. That's fine. So what will your feasibility criteria be for this? How are you going to explore feasibility? What data will you collect?
Yes. As far as in the function category, there is a really nice outcome measure that the therapists at Nationwide, the Low Lab, came out with, and it's called the neuromuscular grow. It's the only lifespan outcome measure we have in neuromuscular disease, and we learned it from the SMA treated population. One of the unique things about that outcome measure is it does a really nice job picking up a change in skill level of the most severely involved patients. So I'm using that, but then I'm also designing my own outcome measure that's device specific.
So that sounds like that's a measure to look at the effect of it, and you can get some early idea of effect, with feasibility studies. So you can look at effect size, etc. But actually, what are your feasibility criteria? What do you think could be the issues about the feasibility of rolling this out? In terms of people using it in their homes, about the caregiver support. So how are you capturing? So because I've done quite a few feasibility trials for rehab interventions, so we often will set what the feasibility criteria will be, because it's actually really important, and actually thinking about what sort of data source would that be?
Yes, we definitely have a qualitative questionnaire for the caregivers, and we also have a qualitative interview that we're videotaping. And then I do a baseline visit in person, and then we do a monthly Zoom visit that if the parents would prefer it to be in person, it can be if they're already here, at our neuromuscular clinic. But that's just an opportunity to listen to how they're using it at home, hopefully be able to see into their home and watch them. And I'm expecting kind of what'll happen. What's currently happening is they say, hey, we noticed this and she wants to use it this way.
So what I'd suggest then is actually thinking about, what you would hope to see like for a really good session, and then maybe thinking about how you might tick that off to be able to see is the quality of the interaction that you would want to happening. And also with the interviews, are you going to do some sort of thematic analysis on them to see what comes through?
We have one person on our team that's done a good bit of qualitative, so she's handling that. And one thing we're really excited about is we have a secure Zoom link for every family. And because this just happens all the time, I'm always getting videos from parents of, look what they're doing. They're so excited to tell about their child's, improvements, that they can actually at any point during the study upload videos of what they catch their child doing at home, if they see an improvement inside or outside of the frame.
So how you then bring that into something that's measurable would be interesting, and that's something to think about for a wider study. But also, how are you going to, because if you think about the feasibility part of that is, do the families engage with them? And of course, you've got some very motivated families now, but how's that going to be across a bigger cohort where there might be variability in what the caregiver circumstances, the home life, etc. So how are you going to monitor engagement with the treatment?
Yes, they will have regular engagement with me. So at baseline, and then every month via Zoom, and then end of study. But then they can also initiate more interactions.
So have you thought about things like using a diary or some sort of,
Oh, I'm so sorry. Yes, they are. I thought I guess my screen's not being shared that has the schedule of events. They actually are keeping a training log.
Good.
And sending that in regularly so that we can have that.
Yeah, lovely. Thank you.
Sorry, Jenna. We're just at, we don't have slides shared at the moment, but no, you explained that pretty well. David?
Yeah, so can you remind me inclusion, exclusion, and age range that you're looking to enroll?
Yes, birth to 5 is the initial age range. We've done the most testing on that size child. We're hoping to be able to expand that though, once we kind of get this first group up and running.
And how about out of the families that you've interacted with, are there people that are not interested in this?
I don't ask them if they want to participate in the study at this point because it's not been IRB approved. We just let them know, we have this product, here's a waiver. Could we try it with you at clinic? We're helping design it. Eventually we want to have a study. I haven't had anyone say, don't call me, but I won't be the one because I'm the designer of the device. I won't be the one reaching out to the families.
Okay, so you really don't know if people are ubiquitously on board with this kind of approach or not.
I think they are from the feedback of I go to their therapist sessions. Yeah. So the feedback from the therapists and the parents are, when's the study starting? When can we get one? I mean, I think the feedback is very, very positive.
Yeah. And then long term, how do you see this fitting in with current approaches? I know this came out of like necessity, which I think we have to look at the positives, which this is clearly a positive. It's exciting. So how do you see this like long term, and right now you're looking at a kind of a more narrow age range. What about the older kids, and how do you see this? Is this a modular device that you think will just like be able to expand with different ages and different ranges of ability? What are your thoughts on that?
Great question. I mean, I used to rig this over NICU beds when I worked in the NICU. So it has grown. I put a 21-year-old in it the other day who weighs 85 pounds. It worked very, very nicely. So we're learning as we go. That would be the goal, is to start with the youngest children in our clinic and then very slowly add older and heavier cohorts. And then ultimately would be able to get enough data that this could go to FDA and not be listed as a device under a more dangerous device number, but have its own number. So that's part of being able to get study data, is to then have it become an actual FDA product and then have it commercialized so that more children could benefit from movement.
And in the larger individuals that you've implemented this approach in, is that something that takes a lot of help? Or is that feasible with like just one individual or one parent, or?
Yeah.
Okay.
Yeah, it's because you can really hook them up very slowly. You kind of get them very comfortable, and the kit comes in a very nice bag that says Explore Land. It's very functional. It's very cute. The moms like it. And then it comes, with all the different parts. And then it comes with another bag that keeps all the different 12 straps and accessories together. And so you just hook them up very, for the small child, you just do it it's through developmental play that you're hooking them up. And honestly, once they see you hang a toy and they can't reach it, and you get one hand up and they start playing, they're only too ready to have the next hand come up and start playing. So I haven't really experienced any of the children to be scared of it. I think they really kind of their personalities come out once they're able to start interacting and playing. And then what's really sweet is they hear their parents praise them and brag on what they're doing. And it just kind of grows. It's a really sweet thing.
So it's all about improving patient care, but also you do have to think about the financial aspect. So what kind of model do you expect this kind of intervention could have? Like, where would a healthcare specialist, a therapist, or a physician fit with this kind of, device? And then it's clearly more than just a device. It's probably going to be some guidance. So I know that's way down the road, but you have to plan that out to get there. What are your thoughts on that?
Yeah, I've actually been adopted by the business school. So I got to go through kind of a market analysis commercialization plan with them that was really helpful. And then, the designer of our straps and cuffs, Fennec Corporation, they only sell to medical professionals off their website. So like a family can't call in and say, hey, I want to order this. I have to call it in for the family. And so we really love that model. To be able to sell with medical professional advice. And as I go and just observe sessions, I have the therapist do their session, and then I just kind of join them. And then I back off. And what I notice is these therapists are so intuitive and the families are so intuitive. And I really try to move myself out of the picture. But, long term, this is going to take a company. It's going to take having a website and having it FDA approved and being able to offer the education when the family gets it that's needed, offer the education for the medical professionals. And you're right, that is a little bit down the road.
I wonder about merging it with some kind of measurement of movement and quantifying movement and range at the same time. So you get both in one, and then you get back and forth to this platform. So then it's both a intervention and a quantitative outcome. And you can say, is there a problem with a limb? Maybe there's pain, maybe there's range of motion issues, and so on. So just a thought. I think you have to really think about the interface.
Yeah, I do have that. So I made a functional outcome measure called the FUN-M, the FUN functional measure, because a child's work is their play and it should be fun. And so it's kind of like you have two clipboards with the same outcome measure. First, you score them without being hooked up, but then you score them while they are hooked up. And you start with, can they bring their hand to their mouth? Well, if you have scoliosis and you're like this, the way you bring your hand to your mouth is here. That's a lower score than can you keep your head in midline, which takes more control, and can you bring it here? And then can you reach one arm, two on? Yes.
We've got about 30 seconds left. Tracy, you did have your hand up, but has it been retracted? Has your question been answered?
Yes, yeah. David sort of asked some of the questions that I had. So
Great. And there is a question from the audience which does link into something I was going to ask around the cost. And I guess some of the, the patenting and intellectual property, I understand you're the designer, Jenna. Why don't you just talk us through in literally 20 seconds, if you could, some of those issues?
Yes, it's been disclosed. UF owns it, and that's as far as we've gotten. It's not been licensed, and we're working on all of the above. Is that a good 20-second answer?
It's a good 20-second answer. Are there any kind of estimate costs just to put people in?
For this study with me making the kits, the $7,000 comes from, if I want to give the child everything I think they would need at the home, it's $350 per child.
Got you.
And that's with me handing it to them, not shipping it to them. And then the other $3,000, as you could see from some of my pictures, these kids are vent dependent and have travel barriers. So it would either be me going to their home or them spending a night at a hotel, and we'd help fund that so they could be fresh when I test them in the morning.
So James, since you opened that door on business development, so is your goal to find someone to license it to, or to do your own startup, or you don't know yet?
I don't know, honestly. Okay. We're just thinking through all angles right now.
Okay.
And there's not a license at this point.
Right.
Brilliant. Thank you very much, Jenna. And again, another fantastic presentation. And you're making the scoring very difficult for us here. But we shall move on now to our last speaker. And sorry, Geeta, I know you did have your hand up there. We will just have to move on just in the interest of time. So our last speaker, Chiseko Ikanaga, talking about some lab work in IBM. Chiseko, are you there? Hi. Just need to share your slides, please.
Yes. Today, I appreciate the organizers giving me the opportunity to present at this Shark Tank session. We start this work to gain insight into the pathogenesis of inclusion body myositis and detect appropriate biomarkers for IBM. We did single nuclear RNA sequencing analysis of muscle biopsies from 7 IBM patients, 3 immune-mediated necrotizing myopathy patients, and 7 controlled, collaborating with Dr. Lucas Schirmer lab in Heidelberg, Germany. He is an expert of single nuclear RNA sequencing analysis. In this figure, each dot represents single nucleus. For example, these clusters show that the expression levels of genes related to T cells were upregulated in IBM. We identified 22 differentially expressed genes whose expression was significantly upregulated in IBM in the clusters of myonuclei related to muscle regeneration and degeneration. We hypothesized that these highly expressed genes can serve as biomarkers to define IBM disease activity, subtypes, stages, and response to therapy. We will confirm that the RNA level is upregulated in IBM using RT-PCR and in situ hybridization. We will also compare the results with that of bulk RNA sequencing of 71 IBM and 44 control samples from NIH, Johns Hopkins, Heidelberg University, and the University of Tokyo. For validating their protein levels, we will do immunostaining and immunoblotting. This is the immunofluorescence staining for ring finger protein 7. This RNA7 co-localized with protein aggregates in the muscle of patients with IBM. At least, the presence of such a differentially expressed gene-derived protein seems to be a good histopathological marker. We also have xenograft model of IBM, and the merit of using them is that they are derived from satellite cells in transplanted muscle biopsies, and the age of each muscle fiber is almost the same, while the muscle of patients with IBM is really heterogeneous in age and disease pathology. using IBM xenograft for RT-PCR and immunoblotting will enable us to assess the myofibers in distinct time points. Actually, we have already done 70% of the AIM-1. I applied for this Shark Tank because the award will enable me to search for serum markers in the AIM-2. This AIM-2 is a little bit challenging, but biomarkers will be most useful when they are detected in the blood of patients. With the hypothesis that the degenerating myofibers will release protein in the serum, we will try to determine the presence of differentially expressed genes derived protein in the cell of patients by immunoblotting ELISA. For promising candidates, we will also use mesoscale discovery and a single molecule array. I think this approach is a candidate gene-based approach. So if this approach doesn't provide us with good serum biomarkers, we will use mass spectrometry as an alternative unbiased proteome analysis. This is what I would like to do if I can get this award. Thank you so much for your attention.
Thanks very much, Chiseko. You're in good time there. Excellent. Who wants to kick us off here? We do have one question from the audience, actually, just perhaps whilst we're bringing our thoughts together around I think you included some IMNM patients, some necrotizing myopathy patients. Just finding the question here, yes. Were the necrotizing myopathy patients on immunosuppression?
Actually, these samples were before starting, we used the muscle biopsy before starting immunosuppressive treatment.
Okay, that makes sense. Who wants to kick off? Dave, I can see you.
I can go. Yeah.
Great.
So I think my big question is, how are you going to determine which are the most promising targets? And you have multiple levels of sensitivity of assays. Is magnitude necessarily going to correlate with the promise of the biomarker? I worry about ELISA being maybe not sensitive enough, and maybe a more unbiased approach. I worry about the bias of choosing which targets are important. So how do you plan to kind of like minimize that risk?
Yes, I think I agree with you, and it is a really important point. I have previously did ELISA by myself, and regarding the sensitivity, I have the same question. So although I told you that I will employ ELISA, if the sensitivity doesn't seem too good, I will employ other methods, including single molecule array and mesoscale discovery. So these assays seems to be sensitive when I refer to other literature. So I would like to use these methods.
How does that fit within the $10,000 prize? If you do have to pivot to a different technology, what are your plans on like applying those funds in the most meaningful way?
Yes. So as I told you, so I have already done single nuclear RNA sequencing and did the most part of immunofluorescence. So if I can get this this award, the merit of if I can get this award, I can increase the number of samples which I use for ELISA or mesoscale discovery and single molecule assay. So the award will enable me to use a large cohort to assess the sensitivity and specificity of these markers I detected.
Great, thank you. I'll hand it off.
Thanks, Dave. I mean, just on that point, and without wishing to sound, confrontational, but it sounds to me like you've already done a lot of the work, and perhaps you could probably just finish it off with the funds you've already got. Do you really need the £10,000 from the MSG?
10,000. So
dollars, sorry, $10,000.
Let me confirm. So your question is whether I really need this award.
Yeah.
Yes. So now in Johns Hopkins, I know that there are more than 200 patients muscle biopsy and therapy. But currently, we are going to use just for 10 IBM samples and 10 controls for now. But if I can get this award, I would like to do the experiment by myself using 200 patient sample. So the award will help me so much.
It improves your numbers and your power to find something quite significantly. Okay, Tracy, could I come to you?
Yeah, I was just wondering about the biomarkers, and when you're looking at these, you know, sort of how are you sort of obviously, as you said, IBM is quite a heterogeneous sort of condition, and you know, how are you looking at that, you know, with regards to correlating that with the biomarker?
Yes, I think it is really important point too. So IBM is heterogeneous. So I think it is important to, for now, for every samples I use, I'm checking AG staining, homolytic staining, and P62 positive aggregates presence and TDP43 positive aggregates or abnormal nuclear clearing. So IBM is really heterogeneous, but I am checking all the muscle biopsy by immunostaining, and so so I have really precise information based on immunohistochemistry and immunostaining. I hope this helps the analyzing subsequent analysis for serum markers.
Mm-hmm. Okay.
Okay,
shall we go to Geeta?
On that similar point that Tracy's made, actually, and again, forgive me if this is a bit of a naive question, as I don't work in your field, but from the samples that you have, what information do you have on the person those samples are from? I mean, is there some, an initial being able to get initial feel for actually what the heterogeneity might be in the sample that, this 20 and then hopefully the 200 if you were to get the award? What information do you have so that you could at least start to make a bit of a stab at understanding how heterogeneous and what factors might be important?
Yeah, I agree with you. It is really important point. Actually, when I came to the laboratory, there are sample lists. But before I use these samples, whenever I use these samples, I try to check clinical information, including, for example, finger flexor weakness or quadriceps weakness, or whether the patient had dysphagia, or whether a patient on immunosuppression or not. So I checked by myself all the clinical information I can collect from the clinical chart. And yes.
Great.
I think perhaps what you were, just to finish off on that point, Geeta, was what you were driving at as well, but this is at least the question I have, is how do you, I mean, obviously in an ideal world, we'd all love to have extra samples. Standing where you are now, how do you know that these extra samples will actually be helpful to answer the question, the research question that you have? And then I'll hand over to Vladimir, who has another question.
So let me confirm. So your question is how this single nuclear RNA sequencing analysis will help clarify the pathogenesis of IBM and detect good biomarkers. So
it's more like you have a number of samples now. This money will help you get some more samples. It's sort of, how do you know that you need more, and that having more will definitely help you to answer the question in a better way than you can now?
Yes. So as other shark person told me, the IBM is heterogeneous. So if I can get a large number of cohorts, although the IBM is a heterogeneous disease, I can reduce the heterogeneity. I'm sorry for my poor English, but reduce the heterogeneity. So the large number of patients will improve the accuracy and accuracy of the assay.
No, that makes sense. And your English is great, by the way. No problem at all. Vladimir, I shall just unmute you now because you have quite a technical question. And Chizeka, you may wish to share your screen again with your methods on.
Yes.
Yeah, Vladimir, you should be able to speak now.
Yes. Yeah, thank you so much. That was a really interesting talk. Yeah, I just have a quick question. So I'm noticing very few dendritic cell expression, relatively few dendritic cells compared to myeloid populations. And I guess I kind of have a two-part question. One is, were you able to express those cells well with single nuclear RNA-seq? Or was that just a population that was difficult to capture? And then secondly, it's a bit striking since, IM-NM should have if anything, an increased macrophage/dendritic cell population signature. And I'm actually seeing fewer, antigen presenting cells based on this data. So if you can comment on both of those.
So let me see. I'm sorry. I would like to let me confirm. So your question is the number. So you are talking about the dendritic cells here.
Yeah.
And yeah. Let me see. So as you know, in previous literature, so in the muscle of IBM, not only cell-mediated immunity but also humoral immunity seems to be involved. So in this group denotes the myonuclei of dendritic cells, and there are some clusters of plasma cells and B cells. So I think this single nuclear RNA sequencing data also shows that the involvement of humoral immunity is apparent in the muscle of inclusion body myositis. Am I making sense, or if it is not what you would like to confirm, I would appreciate it if you could let me know.
I guess it depends on your definition. I mean, we can talk about this later, but I mean, if you go by the literature, I mean, IBM should have an increased number of monocytic dendritic cells, right? And then the other portion of this is fine, even if, if that's the baseline, IM-NM should have a higher number of macrophages and monocytic T cells. I mean, that's part of the pathologic diagnosis, but I'm not seeing those populations in this the single nuclear RNA-seq data, unless I'm reading this correctly.
So this is one of the limitations of this study. So we used the 7 IBM and 7 controlled, but we used only 3 immune-mediated necrotizing myopathy. So if we increase the number of samples, the result may change. But yeah, thank you so much for your for pointing.
Yeah, it sounds like it'd be great perhaps you to perhaps touch base after the meeting. Some pretty good questions there. Just we've got about a minute left. Do any of the other sharks have questions? Nothing major. Perhaps I'll just ask one final question then, just to bring us back to the clinic. And obviously with the lab work and the biomarker work, we always wonder as clinicians looking at patients what it all means really and what difference it might make to them in the future. Where do you see your work going, and how might it translate into clinical practice in the future?
Yes. So in future, if I can find the good biomarkers, it will help. So there are, for example, yesterday there was a presentation of rapamycin and so on. So if I can get the good biomarkers, I can detect the candidate patients using these biomarkers. So we will be able to enroll the more IBM patients in early stages. So I'd like to.
Thank you, Chiseko. That's fantastic. And we'll draw things to a close there. Thank you again for presenting. And I'd like to thank all three of our speakers. I think there were three fantastic presentations, and I'm sure the fellow sharks and the audience would agree. It's a pretty daunting thing to do and stand up here in front of 120 odd people and present their ideas and pitch in such a clear way. My congratulations to you all. And that does bring the presentations to an end. What the Sharks are going to do now is score each of the presentations in a structured way, and as I say, provide some feedback to each of the speakers. And then a little later on, just got the program in front of me now, so actually at 11:10 Central Time, there'll be the announcement of the winner, and then a presentation from last year's winner, Vino, that I think will really sort of demonstrate the momentum that's building now with this prize, and really what this can do for people and their studies. So thanks very much, Rick. I don't know if you have any closing words, or do you want me to just hand over for the break? Rick maybe is not there at the moment. Okay.
I think you can hand over. I think just hand over for the break. Yeah.
For the break. Okay. So thanks everyone. So we've got a break now until 10 a.m. Central Time. This break apparently is sponsored by Genentech, and we shall see you at 10 a.m. For a genetics session, which should be really interesting. So thank you very much again, and see you then.
Thanks, James.
All right, we're going to get started now, back from the break. That was just unbelievably exciting Shark Tank. Thank you, contestants, and thank you, Sharks. So I just going to take a few seconds to introduce our next moderator for this session. We're so glad to have Dr. Laurie Gutman, the chair of neurology at University of Indiana, chair this next section. Good morning, Laurie. Thanks for being part of the MSG again this year. Really appreciate it. I'll turn it over to you.
Thank you. Thank you very much, Rick. It is a great pleasure to be here. I'm really excited about the next session, which is on genetics. We have three really wonderful speakers who I'm looking forward to hearing from. First, we're going to hear about the bane of my existence of VUSs from Tanya Bardakjian, and she is at the University of Pennsylvania. Tanya, I'm going to hand it over to you. Thank you so much for giving this. I'll go ahead and mute myself.
Okay. Hi, everyone. I'm Tanya. Can everyone see my screen? Okay. So I'm going to be talking today about, I think, the bane of anyone's existence who orders genetic testing and the role and importance of expert clinicians in management and interpretation of the variants of uncertain significance. So the dilemma, obviously, is that next generation genetic sequencing has increased the diagnostic yield and accuracy significantly for those of us in the neuromuscular genetics world, but in tandem, that's increased the identifications of variants of uncertain significance. And expert clinicians and genetic counselors are really the key to reduce the burden of this VUS long term. Just to give a little background, multi-gene panels in neuromuscular disorders, some studies have shown up to a 60% diagnostic yield in certain disorders. One large study in a cohort of 25,000 unselected, so not from neuromuscular specialists, actually identified a slightly lower rate of identification, about 20% of diagnostic yield. But what's important here is that that's where the vast majority of genetic testing is eventually going to be, in the general community. And with the 20% identification and the diagnostic yield, you'll see that a good portion of those are copy number variants, which is the CNVs. And with that identification of diagnoses also comes a very high burden of VUSs. 53% in this unselected population had a VUS reported. on their next generation panel. That's 17,630 unique variants to sort through for the clinician. Another example by Rogenbach in 2020 was in ALS, in familial ALS, when they did large next generation sequencing panels. In those that were negative for C9orf72, 35% in familial ALS patients had a variant of uncertain significance. So the burden is certainly significant. If only it were simple, but it's not. So in 2013, the NIH actually funded a pretty large consortium that continues to get larger called ClinGen. And ClinGen is geared towards identifying and ensuring that genes that are said to be associated with disease are well curated. There are panels of experts for every kind of disease category. Currently, they are creating one for ALS. There's one for inherited neuropathies, Parkinson's, many, many disorders, to ensure that it is a well curated, functional input from experts all across the globe so that we can ensure that what we're reporting is accurate. And then, of course, in response to all the VUSs and the discordance among CLIA-approved laboratories for variant interpretations, in 2015, the ACMG and the AMP developed consensus guidelines for the interpretation of variants. Now, I am not going to teach us all how to do that for the purpose of this talk, but ACMG guidelines are what is followed by every CLIA-approved laboratory, and they were designed for Mendelian disorders that are germline in nature with high penetrance and typically small variants. And the key is that the gene must have already a well-established association with phenotype, and that's what ClinGen is working on. Now, this is what the ACMG guidelines look like when a laboratory sees a variant. I'm obviously not going through this, but they have to score based on all of these, based on all of these rows and columns, and under every single interpretation, there's a score that they can give for strong evidence, moderate evidence. It's quite cumbersome, but it is what the labs are using to try to come to a consensus. But even still, labs will come up with discordant results in these 5 categories. So it either has to be benign, likely benign, VUS, likely pathogenic, or pathogenic. Those are the 5 scores that you can see from a CLIA-approved laboratory. Some will include whether or not they think the VUS has a low, medium, or high probability of pathogenicity, but it's always called a VUS. So what are the core questions? Things that we ask are, does the variant alter the function? Is the VUS predicted to have an effect with functional models? What does it look like in large unselected populations? How many people who don't have disease have this variant? Does the variant segregate with family members who are also affected? Functional data can be gleaned from the literature if you're lucky. Simple things like an enzyme assay. If you have 2 VUSs and someone where Pompe is suspected, you can do the simple enzyme analysis. Are there specialty labs that can help? Calpain staining for VUSs, we use Dr. Moore's lab, for example, and we'll hear more about the muscle biopsy for variant interpretation. So we have tools in neurology, thankfully, to help interpret these. Really briefly, what are the core rules with population data? If it's more than 5% allele frequency, that's automatically benign. If it's absent from population database, very suspicious for pathogenic. But we are finding that in the adult onset disorders, complete absence from the population is probably a little bit too strict, because no one knows if those individuals were actually evaluated at the right age. Perhaps they had a condition that developed after they had their genome tested or sequenced for the population database and thought they were healthy, but they actually had some sort of disorder. So we're learning a little bit more about population databases, but they are some pieces of information we use. Segregation or family studies. Is the variant de novo in a dominant condition? Have we proven paternity? Does the VUS segregate with disease? Good clinical information is what's important. And then in recessive conditions, you might have 2 variants, but can we prove that they are in opposite alleles, that they are in trans, which is what would be necessary for a recessive condition? Things like RNA-seq are coming out that we're hopeful that will continue to be helpful. There are some clinical uses for this, but insurance typically doesn't cover it. And in the cancer genetics world, they are being used more clinically as we progress, because we do know it can increase the diagnostic yield. But what do we do at Penn? We do have several genetic counselors in the adult world, and we work in tandem, obviously, with the neurologist, who will perform the deep clinical phenotyping, all the things we know of, and sometimes we'll go back to them, a re-examination. Perhaps I'll say people with this variant should have atrophy of a certain muscle. Can you assess this in any certain way? Either do a muscle biopsy after the testing, it's usually after testing for us, or can we get sampled to look again? And what we've developed is a huge REDCap database. Every individual who has genetic testing, we deeply phenotype, enter their variants, whether they're pathogenic, VUS. We relook at the pedigree. Is there a way we can sort out whether or not this is segregating with disease? We document every single VUS, because patients, their providers in the community will see these results now that there's electronic medical records. We want to document it, and we want to deliver it to the patient to make sure that they understand that a VUS was found and why we think or don't think that it could be causative of their disease. And one study obviously highlighted that there's significant time limitations to this. 46% of genetic counselors report not just going by the lab interpretation and doing further research to try to figure out whether or not even a pathogenic variant is causative of the patient's condition. So to reiterate step by step, we look at the inheritance. Are there two variants in an AR disease? If so, can we prove they're in trans? Is there a CNV or a copy number variant associated? So if a panel was done that doesn't include deletion and duplication analysis, a single variant of uncertain significance, if it's suspicious for that phenotype, you might need to order further testing. What does the population frequency look like? Is the variant in a hotspot? Are there other variants that have been reported associated with disease in that residue? We use ClinVar, which is an international database of curated variants from CLIA-approved laboratories. And of course, a detailed clinical review exam and medical history. Is there anything in the history or physical that makes sense? Just to briefly give you an example, if you've never looked at ClinVar, you literally put in the search engine, the gene, and then every single variant that has been clinically curated or reported by a CLIA-approved lab comes up here. And sometimes you'll see the same exact amino acid, and you will see that there are two different interpretations. So it's very important to look at those, because that's a big clue to us that there's some further research that we need to do. There are international databases like Gene Matcher and Matchmaker Exchange, where we actually enter so that we can find families with the same variant or close by information, researchers, people that are willing to work with us that have seen the same variant that hasn't been reported. Contact with authors that have expertise, clinical evaluations, family study coordination, and of course the REDCap database. We do have a neurogenetics case conference monthly where the neurologists and genetic counselors from across neurology will come together and see if we know anyone with expertise in that field that can give us additional information, referral to specialty clinical evaluators like pulmonology or immunology, if that could give us a hint. And what's really important is if we change our assertion, contacting the clinical lab with our research findings or our new clinical information, reporting by case or series is crucial, because the clinical labs have to follow those ACMG guidelines for reporting, and their info and interpretation is limited by the information that we as clinicians provide to them. So when ClinVar has discrepant assertions, if we find new information through research studies or clinical correlation, or new information is identified in the literature, we are very aggressive about contacting the labs with our information, submitting it for variant reinterpretation, which they very often do. And why is this important because for the patients, VUSs are a heavy burden. Understanding is key to the patient's belief about their risk, their consequent medical decisions, their willingness to be involved in research. And all the studies have consistently shown a mixed understanding of a VUS by patients as well as clinical providers that are not well versed in genetics. It leads to overestimation of risk, anxiety, confusion, and people asking for predictive testing for unaffected family members for a VUS, which is, not really what we would clinically recommend. So the genetic counselor will ensure that the information gets delivered. We assess that, this is ambiguous. It's important to highlight that to the patients. We provide them hope that we will continue to work to reclassify, and the labs do this as well. And the vast majority of VUSs are reclassified as benign. We develop a specific plan, if at all possible, and of course, give that to the patient. And probably the most important is this: we document that there is a shared responsibility and variant follow-up between the provider, the lab, and the patient. We put it in the patient's lap and say, contact us every year or two, because then we can look up and see if there's any new information to be gleaned about your VUS that can help you or your family. I do want to give two quick examples clinically where this really helped. We had a patient who was 25-year-old with progressive dysarthria, dystonia, ataxia. Family history was significant, and then mom had breast cancer at 58, and she had no contact with her father. Exome analysis was done and identified two variants in ATM gene. One was a VUS, one was pathogenic. We couldn't prove they were in trans. ATM causes an increased risk in heterozygotes for breast cancer, and when it's proven to be recessive, it causes ataxia telangiectasia. Mom happened to have the VUS, so this was important for her health as well. We ordered AFP, which is universally elevated individuals with ataxia, and our proband had elevated AFP. Referral to immunology proved an abnormal IG profile. We contacted the lab. This VUS was changed immediately based on our clinical information. And that makes a difference for this lab's cancer panels now. That VUS is no longer a VUS and will be reported and will help people know that their variant increases their risk for breast cancer. And then we had a family that presented to us at 62 with ALS, a strong family history, and 5 years ago had two different next generation panels done. C9 was negative by both labs, but one lab identified a FUS variant as pathogenic and an SOD1 as a VUS, and the other lab didn't report the FUS, but identified the SOD1 variant. The patient at the time was counseled that he has a FUS-related ALS. That's what he's been researching, looking into. His family members, his adult children were counseled that they have a 50% risk. They had decided not to have testing yet, but they did get genetic counseling predictively based on the FUS variant. 5 years later, with new clinical follow-up, the neurologist felt, this really doesn't look like a FUS-related ALS. And when the genetic counselor reviewed the report in the initial genetic test, we realized that it was based on one patient. It was a 2010 reference. It was old, had no functional data. And then the second time it was reported, it was in a patient with essential tremor, not even ALS. And the third report that was used in the CLIA approved labs report referenced the first patient. So it was one patient, no functional data. It would have never passed the ACMG current guidelines. But back then the guidelines had not been established. So we were able also to say that this was very high in the population. ClinVar had differing interpretations, and we called the lab. They reclassified this variant and has been removed from ClinVar as pathogenic and actually removed altogether. And the SOD1 variant now is what is more suspicious for us because it is in a mutational hotspot. It's been reported in several other individuals with ALS, and it's absent from population databases. So for this family, obviously, this is important for accurate recurrence risk, accurate genetic testing and risk evaluation for the family, access to research trials if they become available, and his SOD1 is proven to be pathogenic, will be informative for them, obviously, in the future. So obviously, variants of uncertain significance are a burden in the molecular diagnostic process. They are considerable time and effort has to be spent to assist in the interpretation. And expert clinicians, such as those obviously in MSG, in partnership with genetic counselors, are truly critical to perform further characterizations, evaluations, publications, and to assist the CLIA-approved labs. So research shouldn't just stay research. We should really be supplying that information to the lab so that they can report back in an accurate way and reduce the VUS burden over time. These are just some of the references that I use. I think someone's going to read me the chat questions if there's questions,
Yep.
right, Lori?
I'm here, Tanya. So there were a couple of good questions. One of them referenced, well, first of all, I want to say that your statement of the genetic counselor going above and beyond the lab report has been my personal experience as well. That I think that's, and then that leads to another question that came up in the chat about how important genetics are in neuromuscular disease, and not every neuromuscular group has a genetic counselor. And they wondered, they're asking about your thoughts. I will say that there are, someone asked about neuromuscular fellowships, and genetics is required for the ACGME fellowship. But there's also the question of what if you don't have a genetic counselor embedded in there? How do you do that? So open to your thoughts.
Good questions. I'm not quite sure how to answer that because there is obviously a shortage of genetic counselors, but I do think that the laboratories now do a very good job of providing post-test information and guidance. So where there's considered a conflict of interest to provide pre-test counseling to patients when they're going to utilize their laboratories, these labs like GeneDx, Invitae, which are the molecular genetics labs, use genetic counselors that they provide to help interpret and perhaps guide the management of clinicians. It is a time issue, right? And there are ways that people can learn more about how to do genetics and interpret and use genetics on a consulted basis as opposed to having them embedded, but it's certainly difficult. And what we're trying to work on is actually taking us out of the pre-test world. Just make sure that we can help the doctor order the right test, because that's really key as well, that the right test gets ordered. Triple repeats won't get detected on next-gen sequencing. Whole exome is not the answer for everything. But then we're available more on the back end, because even for genetic counselors embedded in the clinic, the volume is exorbitant. It's just too much to do. We're trying to create pre-test videos that will just explain, you might get an inconclusive result. This is what genetics is going to try to do. And then genetic counselors are available on the back end. But utilizing some of the labs on the after might be a helpful tool for some people.
Yeah, there was another question, but I see that Rick has raised his hand, so we'll let him ask the next question.
So it may be the half-blind leading the half-blind, but since we have such this crazy shortage of genetic counselors, one of the ways that I've been doing it is that we've been putting people put their results they don't know what to do with up on the RRNMF website, and people are weighing in. But again, it's not genetic counselors, it's us neuromuscular docs. But that has been helpful. I'm just getting ready to post one today or tomorrow when I can find the time of a complicated genetic report that I need help in understanding. So that's one way of doing it.
Yeah,
I think so.
Conferences are always helpful. I agree. And that's sort of like a web conference.
Right.
Another question that we had, I know we're going to run out of time, but another question we had was how someone asked what your thoughts were about the fact that most of the databases were heavy European descent. And then a more direct question about what your thoughts were about the Franklin database.
So the Franklin database we don't use very often. I don't have an opinion about it, but we usually find discordant results, which don't really help us. They add a lot to our confusion. The biggest issue with the population databases is what you brought up, and I was very remiss to not bring that up. If you have a patient who is an ethnic minority, your variant rate is very high. And that is a very large initiative now in the genomic space to increase the diversity of our population databases, because it is I think it's over 90% Caucasian, Northern European Caucasian specifically. So that does not help in the general genomics world.
And one last question. So if you have a VUS and the phenotype fits and it's high pathogenic, what's the path for the researcher? It sounds like we need to publish it. Is that correct? Yeah.
Yes. Yeah. So publication number and assessment is what ClinVar will actually pull every publication, whether it's a case report, functional study, and score each one for the variant as well as the gene. And then the laboratories can use those assertions from ClinGen in their criteria, but they need to know that functional or informative information is out there.
Right.
So helping the one patient or two patients is not enough. We must make sure other people get that.
So Laurie, I can guarantee if MSGers want to get that information published quickly or definitely get it in, we can do it in the journal, the RNMF journal, without much trouble.
Thanks. Thank you, Tanya, for an absolutely fantastic drive-by about the VUS.
Sorry, yes.
No, I know we don't have a lot of time, and I know people have tons of questions. I'm just being told we have 1 minute left. So I guess let me scroll back and see if there's some other questions.
A couple more.
Yeah, there are other questions about, most of them are actually kind of linked to what we've already talked about.
Can I just add one thing? If there is a sort of web-based interpretive neuromuscular thing where everyone's working on trying to clarify genetic results, I would suggest that somehow, I don't know much about this, but if there is a genetic counselor or a molecular geneticist that is on there, great. But I think that's a great way to expand our tools. We don't need to see every patient every time, but certainly we might be able to add some pointers on how we can contact the families or interpret the results in a different way.
Yeah. So someone asked what the Franklin database is.
Yeah, it's a type of, it's a database that's geared towards helping with variant interpretation. They use advanced tools and bioinformatic software to try to better give us an idea. It's not perfect. None of the databases there are perfect. So someone just asked what we think about it. It's great. It gives us a little bit more information, but none of these databases are perfect because they often don't answer our question, which requires either functional or clinical proof that that variant is causative.
Great. Great. Again, thank you very much, Tanya. That was really a wonderful talk. And someone actually had asked about the fear of the decreased number of muscle biopsies that get done now and trying to interpret VUSs. There had been some comments about, in actually Tanya's talk about sending muscle biopsies to Steve Moore's lab for specific research studies. And I'm happy to introduce one of my former colleagues from the University of Iowa and a good friend, Kara Jones. Dr. Jones is in Iowa, and she is going to give us this wonderful next step of what do you do with VUSs, and is there an additional tool like maybe a muscle biopsy correlation? So take it away.
Thank you. I'll share my screen. Okay. So thank you to Laurie and the organizers of the MSG for including me today. It's really fun. I actually knew Rick as a MD PhD student at KU, so it makes it extra fun to be here today. And I am a consultant advisor for Audentes, but my work with them is unrelated to my talk today. And the study that I have done here is through NIH funding with our Iowa Wellstone Center. So you already have received a great introduction to next generation sequencing and VUSs from Tanya, so I will be brief about it. But basically, as you all know, this is the way of the future for neuromuscular disease diagnosis. So many companies have their various neuromuscular disease genetic testing panels with really highly varying numbers of genes. You can do very targeted panels, or you can do your MNG comprehensive panel that has 909 genes, which is just mind-blowing. And while it is extremely helpful for diagnosis, if you can get a nice pathogenic variant that fits with the clinical phenotype, you can be done. You don't have to proceed to a muscle biopsy. But of course, if you don't, you end up with a variant of uncertain significance. If you're lucky, one. If you're unlucky, multiple, which is extremely common. And as you were already introduced to this standard from the ACMG and from AMP, really tells the labs how to be classifying variants, sequence variants that they're identifying. And they use all of this information to try to determine if something is pathogenic or benign. And if you're lucky, you get your benign or likely benign call, or pathogenic or likely pathogenic call that can be really helpful. But most of the time now, what we're seeing, especially with the larger panels, are multiple variants of uncertain significance. And this leaves us with significant diagnostic uncertainty for the clinician, for the patient. And there are, multiple ways to deal with this, as Tanya was telling you. So you can look more into familial genetic testing. It's helpful to know if a VUS is inherited, particularly if it's a recessive disease, if it's in cis or in trans. under the age of 18, and an even spread of males versus females. So what do we do when we get a muscle biopsy? How do we try to help interpret this VUS? Well, we have a number of tools at our disposal. And what we do is we look at the muscle biopsy, we look at the clinical history. Hopefully we've got the genetic testing actual report with us and not just a note that says, oh, there was a VUS and 2 genes. And then we try to decide what is going to best help us really interrogate those VUSs and really be able to decide what's going on in this biopsy. The most helpful things in these cases are often our histochemistry, our immunofluorescent staining, our Western blotting, and electron microscopy. So when I looked at data from every case, I wanted to determine how helpful our biopsy workup was. And so if it was diagnostic or helpful, that actually breaks up into 3 categories. We were able to suggest in some cases that the VUS was likely to be disease-causing based on our functional data. Sometimes, which is also helpful, we were able to suggest that our VUS was not likely to be pathogenic. So that was something that really the clinicians could say, okay, we don't really need to worry about it because the protein expression is totally normal in this patient. And this category was the one that actually surprised me. I didn't even think about it until I started doing the study, but there were a number of times where we were able to suggest another genetic muscular dystrophy or myopathy completely unrelated to the VUSs that were presented. And then, of course, the non-diagnostic category was when really our workup was not able to contribute to the interpretation of the VUS. So out of our study, 33% of the time we were able to suggest a VUS was likely to be disease-causing. I'm going to give you an example. This is a 30-year-old woman who experienced muscle soreness and dark-colored urine after vigorous exercise related to her military service in her early 20s. She presented at the time with rhabdo. Her CK was extremely high. And then this happened multiple times after that. I think she ended up getting her muscle biopsy in her 30s. But at neurologic exam, there was no weakness, no sensory symptoms. Her reflexes were within normal limits. So a genetic testing panel was done, trying to determine what was causing her rhabdo. There was one heterozygous likely pathogenic variant in SGCA, the gene that encodes alpha-sarcoglycan. This is an autosomal recessive disease, a form of LGMD. And then there was also a heterozygous VUS in SGCA, but it was unclear whether this was in cis or in trans. There was no parental testing that could be done. So this was a big unknown. She also had 2 other additional VUSs, one in PLEC and one in CINI1. And so a muscle biopsy was done. And what you see in our muscle biopsy is that it's really not obviously dystrophic. We have a little bit of fiber size variation, but overall, sorry, I think this is in the way, a little bit of fiber size variation, but overall, this is a pretty normal looking muscle biopsy, a few hypereosinophilic fibers, but no necrosis or dystrophic features at all. But we proceeded with our testing anyway. We knew we could do testing for alpha-sarcoglycan. We could do electron microscopy if needed. But alpha-sarcoglycan was extremely helpful. So here you can see we do immunofluorescent staining. Here's the control at the top, the patient at the bottom. And there's a significant loss in sarcolemmal alpha-sarcoglycan positivity. When you do beta-sarcoglycan, it's actually fairly well preserved. Whereas gamma-sarcoglycan shows a loss of immunoexpression, but not as significant as alpha-sarcoglycan. And delta-sarcoglycan was also fairly well preserved. This pattern where alpha-sarcoglycan is really significantly decreased with a little bit of a decrease also in gamma is what we always see in alpha-sarcoglycanopathies. It's very diagnostic. So this is a case where our muscle biopsy findings combined with our genetic testing results highly suggest that the variants in SGCA are likely to be disease-causing. So she was diagnosed with a sarcoglycanopathy based on her muscle biopsy. However, you may go back and you may be asking as clinicians, but she had no weakness. She had a muscle biopsy that looked pretty normal and she had rhabdomyolysis. How does this fit? Well, actually, if you dig into the literature, there are 6 cases of patients with primary sarcoglycanopathies that present with this kind of a presentation. It's very rare, but it can happen. And in one of the papers actually talks about alpha-sarcoglycan specifically. When we look at all the genes that we were most frequently suggested to be disease-causing based on our workup, calpain-3 was one of the highest. And as Tanya mentioned, that we do Western blots for calpain-3. And 9 of the cases we were able to suggest were disease-causing. DMD and RYR1 were also highly represented in this category. In the category where we were able to suggest a VUS was not likely to be pathogenic, that was 35% of the time. And this is a case of that would is prototypical for that. So a 70-year-old man with a high CK, proximal muscle weakness, genetic testing was done that showed an RYR1 heterozygous VUS. And this can be an autosomal dominant disease, obviously more often presents in children, but milder forms can present in adulthood. And LDB3, autosomal dominant myofibrillar myopathy, which could absolutely present in someone that is older. The muscle biopsy showed very conspicuous ribbed vacuoles, which I'm highlighting here, lots of fiber size variation. And when you do a trichrome stain, these vacuoles are very nicely red rimmed for the name red rimmed vacuoles. So red rimmed vacuoles can be seen in myofibrillar myopathies, not characteristic at all of RYR1 disease, but we went further to try to continue to further work this up. So we did staining for alpha B crystallin and myotilin, which aggregate in all of the myofibrillar myopathies, no matter what gene is causing it. And there was no aggregation of abnormal alpha B crystallin or myotilin or Desmin. And then we did EM to really try to look for some really subtle cores or rods that you could see in RYR1 disease. And the sarcomeres were very nice and normal, and there were no cores identified. There were no myofibrillar inclusions either, like you would expect with LDB3. However, when you do Cox SDH, a dual stain, blue fibers are Cox negative, and you can see very nice blue fibers here showing up. There are many Cox negative fibers, which is very abnormal even for this patient's age of 70. And TDP43 was aggregating abnormally in the cytoplasm. In addition, when we do MHC class 1 staining, here's the control where capillaries are positive normally, but the myofibers are negative. And this patient had diffuse myofiber positivity in varying amounts, as well as complement C5b9 deposition in capillaries. So when we put everything together, there's nothing to suggest RYR1 disease. There's nothing to suggest LBD3 disease. However, there are diagnostic features of inclusion body myositis, and that is what, in fact, this patient had. It was just simply inclusion body myositis, and the genetic testing was a red herring. When the genes that we were most frequently able to suggest that were not disease-causing were RYR1, dysferlin, DMD, and myrisin deficient patients. The last category that was diagnostic or helpful was this, where another muscular dystrophy or myopathy was suggested, which was 12% of the time. And this is a case of that. This is a 4-year-old boy with a high CK with proximal muscle weakness. He did have DMD deletion, duplication, and sequencing testing, as you would hope in a 4-year-old boy with a high CK and proximal muscle weakness, but it all came back normal. So a larger neuromuscular disease panel was performed, and they found a heterozygous VUS in SGCD, the delta sarcoglycan gene, but that's an autosomal recessive disease. So a muscle biopsy was performed. This is a very nice example of a dystrophic muscle biopsy. We have some early myonecrosis here with some coagulative necrosis of myofibers that are light pink. We have macrophage infiltration here. We have endomesial fibrosis in between with lots of fiber size variation. And my favorite, the grouped regenerating fibers to help suggest a muscular dystrophy. So delta sarcoglycan immunostaining, if you want to start with that, you can see here the control is very similar to the patient's delta sarcoglycan. There may be a slight decrease, but overall this is very normal and does not suggest a delta sarcoglycanopathy, which really should be entirely negative if that were disease causing. However, when we do our full dystrophy panel, what we did identify was that when you stain for dystrophin, the patient's dystrophin is entirely negative, as you can see on the bottom here, with maybe just a hint of some residual positivity. nNOS is likewise also entirely negative, which is very helpful because in patients with Duchenne muscular dystrophy or some forms of Becker muscular dystrophy that affect the nNOS binding site, we see negative staining in dystrophinopathy patients. And utrophin, which should not be expressed in a patient of 4 years of age, is highly upregulated in this patient. And so this is a really interesting finding despite the genetic testing results. So we did not have any evidence of a delta sarcoglycanopathy in this patient. However, we do have diagnostic immunostaining to suggest a dystrophinopathy. which is what we had diagnosed. And it turns out when a research RNA sequencing was performed, there was actually an intronic variant that led to inclusion of a pseudoexon in intron 7 that was thought to be the genetic alteration causing this patient's dystrophinopathy that was completely missed on next generation sequencing, as it would be. When we look at this category, the dystrophinopathies came out as the most common disease that was suggested that was not listed on the patient's next generation sequencing testing results. And then 20% of the time, our workup was non-contributory. This is cases like collagen 6 diseases in adults where you really don't know whether a positive immunostaining can be seen in patients with collagen 6 disease or not, things like that, where really what we did really didn't help. So in conclusion, muscle biopsy testing can really give you supportive information. So we were able to give helpful diagnostic information in 80% of the cases. And the most common genes that were reported as VUS were collagen 6 genes, RYR1 and CAPN3. The most common cases that we solved were calpain-3, dystrophinopathies, and RYR1 disease. And dystrophinopathies were our most commonly suggested as our other genetic muscle disease. And I have to thank all of the people that were involved in our study here. Of course, Steve Moore, who's been my mentor, good friend, and colleague here, and our Iowa neuropathologists, all of the referring neurologists and neuropathologists, some of you are on today. I really appreciate you sending us cases. And our Iowa neuromuscular colleagues, a strong group. Lori's here, even with her star, because she's no longer at Iowa, but I still include her in the group here because the group here is really what makes it special. And of course, our patients and the people who've helped with the work. And I'm happy to take any questions.
Thank you so much, Kara. That was great. I wanted to say that whoever is writing on the screen, I don't know if you realize, but your writing is showing up on the main screen. So please try to avoid that. I'm not even sure how you're doing that, but that's my technical. I'm too old to know that stuff.
I thought the speakers were doing it, Lori. I thought the speakers were doing it.
No. So there were a couple of great cases, questions that have come up already. So a really wonderful presentation, Kara. One of the simple questions, or maybe simple questions, was about if you have an old muscle biopsy, and is it feasible to get leftover tissue sent over to you guys for immunohistochemistry testing?
Yeah, absolutely. If there's, pretty much everything we do is going to be on frozen tissue. Of course, EM, if you have tissue and glut, or EPON embedded, we could do that too. But yeah, frozen tissue that's even extremely old. We can, tissue from the 90s, early 2000s, even the 80s. We've been able, if it's been frozen well and preserved well, we can absolutely do immunostaining on that, and we can start out with just something to make sure that the proteins have been preserved before we proceed with anything more.
Okay. Dr. Hanna, you had a question?
Yeah, thank you for a really, really great talk. It kind of demonstrated just how labor intensive it is to actually go deeply into the histopathology to understand these variants. Do you think there's any potential, or what potential is there for automation of any of this using artificial intelligence approaches? Not that neuropathologists aren't crucial, but
yeah.
We have, as you say, thousands of VUSs, increasing genetic testing, lots of uncertainty, and it's clear biopsy can be really helpful, but can we automate it?
Yeah, I mean, I think that there are, if you look at some of the clinical trials, they are trying to use automation to analyze immunofluorescence staining. So if you, I could imagine if someone wanted to create, programs where we could do the staining, and then instead of a neuropathologist looking at it and saying, yes, this looks decreased, a computer AI system could potentially do that. Yeah, absolutely. I still think that the decisions that need to be made about what testing to do and how to proceed with that, are best made by a clinician, by a neuropathologist.
Yeah,
And
yeah. No, thank you. It's just the volume issue that may need some thought. Thanks.
So a couple of other questions that came in. One was from the referral muscle lab at Mayo, asking if you have any suggestions on how to get better phenotype, clinical information, including phenotypic and labs, et cetera. Do you have like a particular form that you have people fill out, or how do you get all that data?
it's really hard. I mean, that's been a huge problem, is that you get a muscle biopsy and sometimes there will be literally no history with it. So we kind of fly blind. But yeah, I think that we call. Sometimes I have people here, staff that help me call. Sometimes I make the phone calls myself and just hound people. There is a form and it has all of the important information, but no one ever very rarely does anyone ever fill it out. Unfortunately, that's just the way that it is.
Yeah. And another question specific to the IBM case, there was a question about whether or not the patient was re-examined and had a more typical pattern, like finger flexors, and because of the concern of LGMDs that can pathologically look like sporadic IBM.
Yeah, so they actually, that case came from, I think it was from UC Davis, and they presented it actually at one of the AAN meetings. So once they went back, the clinician that was currently seeing that patient went back, she said, oh yeah, this actually seems very much like sporadic ABM, and I don't really know why genetic testing was performed, so yeah.
So there were a couple, someone specifically asked, Dave Lacomas is asking about, do you evaluate for anoctamin or Titin?
So we still don't have an antibody for anoctaminopathies. We've been trying to get one worked up. That is a really, really tricky one, so we don't. But I will say we've seen that those patients do have a lot of sarcolemmal complement, so I can suggest it if I'm seeing a lot of sarcolemmal complement in the right clinical context. Titin is another tricky one, although I do think I have a biopsy right now on my desk that I think is a centronuclear Titin myopathy. So it can be done if it's a Titin and it fits, with one of the histologic phenotypes, but there's nothing specific for Titin, unfortunately. That is a really tricky one too.
Tony Amato says his experience is biopsy and immunohistochemistry yield is about 30%. They're looking into muscle transcriptome testing. So
very cool.
Yeah. And someone asked if this will be published. Kara?
Yes. Yeah. We're working on it now. I'm trying to add in a 5th year. So once we get all the 5th year data all combined, then we're going to get this out. Yeah.
Yeah, the concern being, how do you get insurance companies to pay for further diagnostic testing once you have the genetic testing done?
Yes.
So, yeah. And how is it? So there's about a minute left. So I don't know if anybody has any further questions. I guess, what is the best way to get biopsies to your lab if people are interested in that?
Yeah, so they can feel free to. I'm easily found on the Iowa webpage. You can email me, but you can just go to the Iowa Diagnostic Labs, and there are requisition sheets and things like that you can send. Absolutely, we're happy to see any biopsies anytime.
Great. This is such a fascinating correlation study. I really appreciate that you put it together and have pulled in all this interesting data. Also, it's helpful, when you, as a clinician, when you feel like there's something there, but it just isn't. The VUSs that you get back are not even like close to what you'd expect. So
yeah, this has been it's very satisfying for us too to find diagnostic and helpful information in the muscle biopsies for you all too. Yeah.
Well, great. Thanks. Kara. Nice to see you. Great. Now I get to introduce another good friend. So I'm really pleased to introduce our last speaker for this session, and that is Shauna Feely, who is also at the University of Iowa and probably has taught me more about neurogenetics and neuromuscular diseases than anyone single-handedly. And I am a difficult learner. Tons of patients. So figuratively and literally. So I will let Shauna go ahead and talk to us today about genetic testing in neuromuscular disorders and some of the ethical and social implications of that.
Hi, everyone. Can you see my slides okay? Okay. Let's see. No, exit, then stop share. And did that fix it? Okay. Thank you, Lori, and for the group to invite me today. I have no disclosures. I've been a genetic counselor in neurology and neuromuscular disease for over 15 years. So I'm just going to talk about what we're talking about in terms of genetic testing when we offer this to patients, and interpreting, as we've talked about how difficult interpretation of genetic testing can be and how we prepare patients, communicating uncertainty to the patient, pros and cons of testing, and some case examples. So in general, there's lots of different times when a patient may be approached in terms of testing. That can include newborn screening, carrier testing for couples who are planning family, predictive testing for people who know that there's something in their family and are wanting to find out if they're at risk. Diagnostic testing, of course, which was the focus of a lot of these talks in terms of trying to identify the genetic cause of someone's symptoms. And then prenatal testing or pre-implantation testing, which is used again for family planning options. And there's lots of different types of genetic testing that we've been talking about. Single gene or targeted variant testing to try to sort of narrow your approach of testing if you know there's a variant in the family, or you think you can narrow it down to a single gene. There's multi-gene panels that can range in size using next generation sequencing. And then it can get larger to whole exome or whole genome. Of course, chromosomal testing. And then also there's non-genetic testing tests that can be used to aid in diagnosis. So for example, if there's a family history of CMT1A that is known, you could use an EMG to see if there's slowing on nerve conductions, and that essentially would give a person a genetic diagnosis. So it's sometimes helpful to think of this as sort of a spectrum of testing where you can try to narrow testing as much as possible if you think that you can try to pick up a genetic hit on just a few genes or a single gene, or you can get broader into these multi-gene panels, and this can extend into whole exome or whole genome. Whole exome being 20,000 genes that we're talking about, which is 1% of the genome. And when we're talking about whole genome testing, it gets into a little bit more murky waters because, of course, we don't know what all of that information means. And the more you test, of course, the more you might find, but we don't know how to interpret that information. So sometimes larger is not always better in that context. As we were talking about, there's lots of different labs that are now available for testing. Labs differ in cost, what insurance is accepted for coverage, genes that are included on their panel. So every lab has a sort of a different set of genetic genes that are included, some that are solid in terms of knowing that they're disease causing, some that are more kind of suspect genes, but we don't know if they have a different role in disease. And the number of genes are also different from panel to panel. So these panels change from year to year based on publications and how the labs interpret those publications. And genetic counselors can help people living with genetic disease and the providers navigate this process. So when we're talking about genetic testing for patients, we try to prepare them for all different outcomes. That could include a positive genetic test where we identify a pathogenic variant, it matches their phenotype, and we know that that's disease causing. Some labs also on their report will say pathogenic variant or positive, even if that person is just found to be a carrier. So it's important to read those lab results carefully to make sure that even when it says positive, that you know that this, that they have the number of mutations that they need or variants they need to have a disease-causing mutation or condition. Negative results mean no changes were found, and the report did not find a cause to the disease. And then, of course, we were talking about variants of uncertain significance, which is a huge challenge, but something that we try to prepare patients for because additional steps may be required in order to interpret that. So, for example, in this case, this was a woman presenting with CMT, and in doing a peripheral neuropathy panel, we were able to identify a genetic cause in GJB1 causing CMTX. But there was also all of these other variants, including one in GLA, which pathogenic variants in GLA, as we know, can cause Fabry disease. So this could have implications potentially for her or her male family members. So it's not enough to just sort of say, okay, well, we found the disease-causing mutation. We also have to investigate what do these mean and what impact and what follow-up studies may we need to do in the patient or family members. We can see here that this is showing that there's no male-to-male transmission in the pedigree. So this would be consistent with a CMTX, but we would also maybe offer family testing for Fabry or looking at studies that we can do to check for patient or male members of the family to make sure that's not in play here. So we know the variants of uncertain significance are a common problem. Reports show that 83% of patients have variants that are rare or of uncertain significance. And so it's something that we have to manage in the genetics area. But we also have to remember that just because something is rare in there doesn't mean it's disease causing. And so there's a lot of work that has to be done to ensure that we're giving accurate information to patients. And Tanya went over a lot of those, which includes segregation analysis, expert analysis, like talking to the experts, looking at databases and literature searches, functional studies, if you can do that, looking at predictions, and then also looking at your own expertise and bringing that into the mix, because, as neuromuscular experts, you can look at these variants and determine whether this matches what your experience has been for these conditions. So this is the guidelines that we use, again, for interpretation of these variants. And it's important to remember the impact that you have when you're communicating with the patient in terms of how you're describing these variants. Language that can overemphasize the variants, such as this variant may be the cause of your symptoms, could impact the patient negatively, and then that they start to attach themselves to that diagnosis. They think that that's the cause. And so we have to be careful about what language we're using and expressing that there is some level of uncertainty and that more information needs to be gathered before we can really tell a person whether this is the cause of their disease or not. You have to be as certain of this as that this is the genetic diagnosis, that you're comfortable with patients making family planning decisions based on that information. So a person might, for example, terminate a pregnancy based on that pregnancy being affected by or having the same variant that they have, even if it's uncertain. So we have to really be definitive in knowing that this is disease causing. And like I said, patients get attached to the name of their diagnosis and their genetic information. So it's important to be careful with this word choice. So some lessons learned are reading lab reports carefully and doing your own investigative work to determine if you concur with the findings. I've had a lot of experiences where patients have been sent to us, and they may have a heterozygous variant in a recessive disease, but they've been told they have that condition, which then you're taking away a diagnosis. I've even had just recently a case where there were multiple variants in the child that was affected. They appropriately did segregation testing in the parents, but the parents, the father who was found to have one of those changes was told he had passed on that condition to his son, even though it was actually a different variant that was de novo that is probably the cause of the patient's condition. And so it brought the father to tears that he had not essentially done this or caused this in his son, but he had been living with that guilt for some time. When variants can't be classified, going back to the family history can also help determine risk. And so we encourage people to go back to the family history and assess risk based on that. They recommend that people reassess variants annually. So it's not just like a one and done situation. More reports and things are coming out all the time. And so, yeah, even when you get that report, it's labor intensive, but who does the reassessments of these variants and when? Is it just when the patient shows up in your exam room, or are we obligated to report that back when we have new findings? Careful phenotyping and your expertise is vital to variant interpretation and muscle biopsies and all of this type of testing, as we've learned. And reporting your variants is vital also to moving forward and our knowledge moving forward. Some of you may have seen the impact in your hospitals about the 21st Century Cures Act. This requires hospitals and labs to result out their reports as soon as they're available. There's a potential impact in terms of patients who are, for example, going through predictive testing and learning at home that they're positive for a genetic disease that's in their family. And also, genetic testing, as we know, can be easily misinterpreted. So that is another potential for harm. And so there's a lot of hospitals that are kind of dealing with the way they're navigating genetic testing, but also other types of testing in specialties like oncology and things like that. Genetic testing is not always the right choice for everyone. So as genetic counselors, we kind of go through pros and cons of testing. Some of the pros are to determine if other family members are at risk, family planning, possible access to treatment trials or research or treatments if available, and having a definitive diagnosis is just really important for some people who've lived for a long time with a debilitating condition. Family planning options include those that are to have your own biological children while not passing on the genetic disease. There's also different types of donation or adoption programs like donors, sperm or egg, embryo adoption, where you still carry a pregnancy, and then traditional adoption with a range of fees that are associated with each of those choices. Cons for genetic testing may be that there's inconclusive results, and what does that person do then? The emotional impact on a family system, if positive or negative. Sometimes we deal with folks who test negative and have survival guilt because of that. There may not be a genetic testing available, and then we're going into research-based testing, and what that means and how long it takes can be different from patient to patient. There may be concerns about genetic discrimination. So a lot of conversations I have with patients are about what protections exist and what don't, where the kind of holes are in those protections. And then cost, and we do help patients navigate towards a lab that will cover most of the testing based on their insurance and things like that. There's other issues that come up with genetic testing, such as when or how do you test children, especially those that are not affected yet with something that's in their family. So, in general, we shy away from testing kids who are asymptomatic, and it's not going to change their medical intervention. And non-paternity can also come up in doing testing. So things that we discussed during a genetic counseling session include information about GINA, which is the Genetic Information Non-Discrimination Act. This protects people who are at risk for genetic disease, and it allows them to know that their employment and health insurance can't be impacted by being at risk for a genetic disorder. It does not protect against life insurance, disability insurance, or long-term care insurers from using that information. So if someone tests positive for a genetic disease, even if they're not symptomatic, they likely will not be able to get any of those types of insurance. It also doesn't protect against people who are already manifesting a condition. And there are major exceptions to GINA, including people who are in the military, veterans, people who are Native American, and then also businesses that with fewer than 15 employees. HIPAA is our protector for people who are affected by a genetic disease. So we again point out to patients what HIPAA means and does, because it's not just about privacy, it's also to allow people to transfer to different insurances, and there's a portability part of HIPAA. But the problem is, again, there's no protections for life insurance, disability, or long-term care insurance. So some cases that come up when we're talking about ethical issues in genetic disease and genetic testing are, for example, things on the newborn screening. There's a lot of debate about what should be included in newborn screening. And we know Pompe disease is kind of one of those that's been heavily debated. It benefits people because of our early identification of babies at risk, and that diagnosis can lead to treatment earlier. It allows families to know that they have a risk of having a child with Pompe and allows for that family planning. And it allows reassurance for families who get their infant gets tested and they don't have a risk for Pompe. But the risks are that all babies with low GAA activity may not have Pompe disease. It doesn't predict what age of onset. So essentially you could be doing newborn screening for an adult onset disease, which a lot of patients are going to have an adult onset disease. And then you're risking things like their ability to get those types of insurances that we described, and increasing potential anxiety about the future and what that means. So just to highlight a case of a family that we went through this process in terms of the pros and cons. This was a family with ALS, so 54-year-old male with a recent diagnosis of ALS, a paternal family history. And due to his relatively early age of onset and family history, we discussed genetic testing options. So here you can see his pedigree with a paternal family history. Father didn't have it, but because of incomplete penetrance, that's not unusual. And through the genetic counseling session, this person actually wrote out sort of a T-table and wrote pros and cons. Pros being that the genetic counselor determined that the insurance would cover the test. It could help detect, why they had a family history of ALS and their diagnosis. It would be able to understand potentially, a better understanding of the course of the disease, and there would be able to use for family members if they wanted to understand if they were at risk. Cons being that the testing did not change the diagnosis, it didn't change the treatment. The impact on his family would be significant. He did not want to know if he had given his children this disease in his lifetime, and even if one of his family had the gene change, it was not going to mean that they necessarily would show symptoms because of incomplete penetrance. So this person decided not to do genetic testing. Other options that the genetic counselor talked about and was, for example, DNA banking, which is an option where that person could have a sample taken, and then in the future, even with that person no longer there, if they passed away from their disease, the family could use that to decide if they wanted to do testing in the future and would still have access to that information. In terms of the context of genetic testing, more and more patients are asking about that. In part, that might be influenced by labs that are offering direct-to-consumer testing, which are becoming more frequent. And with direct-to-consumer testing, people or patients can request easily to transfer data to platforms where they actually can try to harvest or investigate their own data, interpret that. That has a pretty big risk for harm as well, because they obviously might misinterpret that data, and the accuracy is difficult depending on what platform they used for testing. So many of you may know genetic counseling in practice. We help to interpret a lot of genetic testing results and guide patients through the testing process. I've worked in neurology for my whole career, and there's lots of genetic counselors interested in the field of neurology. So it's been growing from 1% of genetic counselors when I started, to doubling in digits now. So I would just consider working with a genetic counselor in your practice, because it can definitely help with patient care and also with the challenges of interpreting variants.
Thank you, Shauna. So one of the first questions is about the GAA benign reportable variants that lead to pseudodeficiency on enzymatic testing. Wondering if you could talk about that a little bit. How do you explain that to patients who might have completed family planning already?
Yeah, a lot of these are challenging, and I think, but trying to explain things in lay language and working with the patient to sort of explore additional testing options that might be appropriate. It's really a case by case situation, but the problem is as these conditions go on newborn screening panels, they're done at a population level, usually without a genetic counselor involved. So as Tanya said, a lot of times genetic counseling is becoming involved post-testing, which can lead to a lot of anxiety and confusion for patients. So making those referrals as soon as possible so that people can interact with a genetic counselor and get correct or information in a way that they might understand. Another question was about the ALS patient with familial ALS. Wondering if he'd been counseled about eligibility for clinical trials, if there was a specific gene. I know you've personally watched you address this before, so I'll let you answer. Yeah, I'm part of my role is I'm 50% clinical genetic counselor, 50% research. So I'm pretty used to explaining different research options and getting patients enrolled in research. So yes, research is always discussed with patients in terms of even participating in genetic studies to identify novel genes to clinical trials. But of course, unfortunately with ALS, the longevity is not, it's terminal disease, obviously, that has a short duration. And so even with clinical trials and things like that, it may not extend life to the point where that person would want to participate, and they may feel like overburdened by the concern that then their children may have a genetic disease. We also talk about, in terms of ALS, in that context of testing, if they pass away before the results are identified, who would they want to have that genetic information reported to, and things like that. So there's a lot of, these conversations are detailed and extensive in both the clinical and research fields.
There are a couple of questions about GINA, and one is specifically about, can you talk about veterans and GINA? And then the other was a specific, have you personally had patients who were denied disability, long-term care due to a genetic result? Yes, I've had people denied. Life insurance is the main one that people can't get if they're gene positive for what's considered a life-shortening disease. And that just is determined by the insurance company and the people that they have on their advisory board. So even if a provider can say, well, they have this disease, but it's not, they may have a full lifespan, they may not die from that.
they can look at, they have their own way of assessing whether they consider something a terminal disease or a disabling disease where that person may not be able to work anymore. So, we've had a lot of well-meaning providers just in the general community, like, primary care providers and things like that think they're helping their patient by just running a genetic test for something that they're at risk for. Unfortunately, that patient is then not prepared for the financial or planning problems that come up in terms of what the implications are of that testing, and it is already in their medical record. In terms of policies like GINA, the reason that they don't apply for people in the military and veterans is because there's a kind of a different criteria that they measure those risks for, especially if they're going to impact, for example, people in the military's ability to do their job. And there also are other protections that are in place that are not don't apply to the general population, but GINA does not apply to those populations.
So along those lines, someone asked specifically about if you'd had a vet with ALS that chose to get testing and that changed the care for them at the VA. I don't know how much experience you have with that specifically.
Yeah, I mean, it's the VA system is, I've worked for them earlier on in my career. They have their own sort of internal system of how they decide treatment plans or care, including genetic testing. Usually genetic testing is run through the genetic counselors that they hire. They have, like, in terms of neurological disease, they have a couple of genetic counselors based out of Utah that help determine genetic testing for the entire VA system. And that usually has to get run through them and approved. And then they do sort of virtual genetic counseling for that process. So that might change over the course of time, but there are different approvals that the VA has for how they do testing and how they manage that care.
Okay. We have about a minute left. Someone asked, is it ethical for patients to buy insurance? Do you think it's ethical for patients to buy insurance before they're about to undergo genetic testing?
I do think it is ethical to do that. I think it's something that helps protect them in their future. We are different in terms of how we do insurance here, because there are some protections for example, health insurance, but not for the insurances that I mentioned. But in other countries, for example, they have a cap so that if you know that you're have a risk for a genetic disease, you can always get those types of things like life insurance and things like that, but they have a cap of a dollar amount of what you could get. So it prevents people from taking, many million-dollar accounts that when they know they're going to die from that disease. So that would be another approach that we could take in this country, instead of just, putting the burden of on patients who have genetic disease. There are even insurances that won't provide like life insurance and things, even if they have a family history but haven't been genetically tested. So, even if someone doesn't get genetic testing, they're still penalized. And there are some reports that people will prevent people from getting access to those insurances if they've seen a genetic counselor, and that involves care and stuff like that. So I think that people should have access to those types of insurance. And it's challenging because depending on the person's life situation, they may not be able to set it up before they want testing.
Thanks so much, Shauna. That was great. And thanks to all three of our speakers in this session. I really appreciate all the input and education that we were able to get out of this session. A Zoom round of applause for all three of the speakers. So, and now I'm going to hand this over to James Lilliker, who will, I think, talk about the Shark Tank winner.
Hi, yeah, thanks, Laurie. Hopefully, I'm coming through. Yes, it's my pleasure to deliver some good news, which we all sort of need at the moment, I guess. And I'll just start really with saying some thank yous, firstly to the the MSG for their generosity in providing this award, and then also to each of our speakers, all three of our speakers, who I think delivered some really fantastic presentations this afternoon, and I hope you found it all entertaining. And then finally, thank you to our Sharks for putting in the time there to score and give sort of very fair opinion and hopefully some really useful feedback to our Sharks. So anyway, moving on, I'd like to move to the next slide and announce that our winner is Katie Dodd with the title The Innate Immune System in Myasthenia Gravis. Sorry, Gil. I spoke over Gil's sound effect there. Sorry. And just to make sure everybody knows that I did recuse myself from the voting of this particular person because I am from the same centre, but the committee agreed that Katie did deliver the best presentation. So thanks very much, and we do look forward to hearing from Katie next year when hopefully we'll hear some results from her experiment. Katie, you.
Yeah, thanks very much. No, I really appreciate it. And yeah, look forward to coming back next year to tell you my findings. Thank you.
It'll be a pleasure to have you. And I think Rick was saying that because you're British, you get the award in pounds. Is that right, Rick?
Well, that's to be negotiated. I want formal acceptance speeches. I think we should have an Oscar type acceptance speech. So next year, Katie, when you give your talk,
I need to thank everyone. Yeah, thanks very much to the Sharks, though, and thanks for being very kind in your questioning.
Congratulations. And actually, I thought all three of them were amazing.
Yeah.
Really Any good. One of them could have been funded. So that was a tough call, Sharks. But
it was tough. It was very tight on the scoring.
Great job. Yeah.
And we'll move on then to introduce last year's winner, Vino from UCL, who has already made, I understand, some significant headway in her project after having been awarded the MSG Shark Tank Award last year. So I'll hand over to Vino now so we can hear an update.
Fantastic. I'll just share my screen. Okay, can you hear and see my slides?
Yep.
Fantastic. Okay. So I'm Vino. I'm a clinical fellow at Queen Square in London. And so I was fortunate enough to win the Shark Tank last year for MEND, which is a clinical trial looking at mixilotine versus lamotrigine in non-dystrophic myotonias. So non-dystrophic myotonias encompasses myotonia congenita, paramyotonia congenita, and sodium channel myotonia. And they are a group of inherited conditions, neuromuscular conditions that cause myotonia, which is delayed relaxation of skeletal muscle after voluntary contraction. And it causes stiffness, but also pain and fatigue and disability and morbidity in a very young cohort. Currently, we've got no cures, but we do have symptomatic treatment in the form of mixilotine. And Statlin and al. Showed that mixilotine was very effective for reducing myotonia. We've been using mixilotine for quite some time now. But there are some limitations to mixilotine. A review published in JAMA in 2015 showed that a significant proportion of patients had side effects to mixilotine. So actually 52.4% of patients reported one or more adverse events to mixilotine. The most common side effect was dyspepsia, but also headache and palpitations. And actually, 19% of patients were non-responders to mixilotine. And patients who have CLCN1 mutations, chloride channel mutations, require a higher dose of mixilotine to have an effective response. So there are some limitations in terms of side effects and non-responders with mixilotine. So more recently, lamotrigine was looked at in terms of efficacy for reducing myotonia. And lamotrigine works in a similar way in that it works on sodium channels And this paper was published in Brain in 2017, looking at 2 different doses of lamotrigine. So this graph A on the right here, so the third bar here is 150 milligram total dose of lamotrigine, and the fourth bar is 300 milligrams total dose of lamotrigine. And they showed efficacy in reducing myotonia using the MBS stiffness score. So it's the myotonia behavior score, which is a 5-point stiffness score. And so there's a dose-dependent effect, so both at 150 milligrams, but more so at 300 milligrams, they showed that there was a reduction in the myotonia behavior score and reduction in stiffness and myotonia. And in fact, they terminated the trial early due to efficacy. And they demonstrated that there was a reduction in the MBS score by 29%, with a number needed to treat of 2.6. Then looking at side effects, so the most common side effect was headache, with 11 patients having headache, but actually 6 patients also had headache in the placebo group. And then 3 patients had a rash. And actually in other literature, so looking at the lamotrigine literature in epilepsy and also in mood disorders, a skin rash is the most commonly reported side effect, and in the majority of patients, the rash stops quite quickly if you stop lamotrigine. But in a very, very small subset, so a 0.01% of patients, the rash can be nasty and cause Stevens-Johnson syndrome. So rash is the side effect to look out for in lamotrigine. And then there are other factors to think about. So mixilotine can't be used in pregnancy, and myotonia worsens in pregnancy. So, that is an issue. Whereas in with lamotrigine, there are clear guidelines for prescribing lamotrigine in pregnancy. Mixilotine we don't use in patients who have significant cardiac morbidity because there is a risk of, or there is a theoretical risk of arrhythmias. We can use lamotrigine in people who have heart disease. And in UK and in Europe, mixilotine is now very expensive, and we can only prescribe it in specialist centres, whereas lamotrigine is very easily available, very cheap, and can be prescribed by GPs. So, there are pros and cons to both. And so we designed a head-to-head trial to compare lamotrigine to mixilotine in non-dystrophic myotonias. So the trial is a head-to-head crossover trial. It's double-blinded. Patients are randomized to either lamotrigine or mixilotine, and they're on either treatment for 2 months in a dose escalation, as you can see here. And then they wash out the medication for a week before they then start the alternate treatment, again, escalating the dose over 2 months. The trial is powered to detect non-inferiority. So we're trying to show that, or asking if lamotrigine is non-inferior to mixilotine. And the primary outcome we're looking at is the interactive voice response diary, the IVR diary. So the IVR diary is a 9-point score. So 1 is minimal stiffness and 9 is the worst stiffness patients have ever had. And they'll record a score daily. And we're also going to record as a secondary outcome measure the myotonia behavior score, which is a 0 to 5 score. And that's the score they used in the lamotrigine trial. And so we'll record that as a secondary outcome measure. We're also going to record degree of activity. As we know, degree of activity interacts with myotonia. And so we're going to account for that in our statistical analysis. Other secondary outcomes are the timed up and go, the time to sit to stand, pain scores, fatigue questionnaires, and quality of life with the SF-36. And we'll also record side effects. So we designed the trial and submitted it or presented it at the Shark Tank last year, which has really, really helped us a lot, and also submitted it as an MSG trial, which has helped a lot. So MSG has been really helpful in the trial design and feedback. We particularly with using the IVR, which is the primary outcome. So that was a score that was validated by MSG, and also with Professor McDermott, we decided that was going to be the best primary outcome measure for the trial. We've had support in terms of statistical support with Professor McDermott, and recruitment and collaboration. So, colleagues from MSG have been, so James Lilliker, for example, has referred patients to us in the trial. So it's been really helpful having collaboration with colleagues in the MSG. And the $10,000, not pounds, has been very helpful for our trial. So with COVID, we've had a lot of challenges, and one is that we've had to have visits done virtually and had to accommodate for patients' reluctance to travel throughout London. And so we've minimized visits. So after randomization, for example, patients don't come to pick up their medications. We courier the medications to them. And actually, couriering medication is very expensive. The medication has to be temperature monitored until it gets to the patient. And so the $10,000 has gone a long way in terms of paying for courier costs to get medications to patients. Similarly, because of patient reluctance to travel with COVID, being able to reimburse patients' travel has been really helpful for our trial. The trial is a double-blinded trial, and setting up the trial, it has been very expensive, particularly with over-encapsulation of the tablets so that they look the same, so it's blinded. It's actually a very costly process. So MSG has been really helpful, both in terms of support and design, collaboration, as well as monetary support. So far, we got the trial up and going, and we opened to recruitment the 11th of August. And so far, we've recruited 14 patients. And we have had challenges, particularly due to COVID delays. The trials related to COVID were prioritised, and so we had a lot of delays in terms of opening to recruitment. We've had to do visits, as many visits as possible virtually, and that does create challenges, particularly the timed up and go, and some of the sort of more objective measures are challenging to do virtually. The timed sit to stand is quite easy to do virtually, but the timed up and go is difficult. And doing things virtually, as I said, has added costs. But we have a very motivated patient cohort, and great collaborators. And so we are recruiting well and hope to achieve our target of 50% recruitment by April next year. So thank you very much to the Muscle Study Group and the team and all of our patients who have been involved. Thank you very much.
Thanks so much, Vino. It's a great presentation, and to get a clinical trial done in the context of PhD is really quite impressive. So well done, and yeah, congratulations again on winning the Shark Tank Award. There's one question come in, and we've probably got a minute or two to take some other questions if there are any from the audience. Tara Jones asks, do you worry that having two separate stiffness scoring ranges will affect the results?
That's a good question. So we explained to the patients very clearly the scores and how to score them. So I don't imagine it will. They're different scores, and we explain it to the patients, so it shouldn't do. Yeah.
So this is Rick. So they're on the IVR. Are you doing the 1 to 10 like we did in the original study? But then you have this other score that's 1 to 5. Is that the question?
Yeah, that's right. So we're doing the IVR, which is the 1 to 9, and then 0 to 9.
I'm sorry, 1 to 9. Yes.
Yeah, that's right. And then the MBS is 0 to 5, and it's a secondary score just because they used it in the lamotrigine trial, and so it allows us to sort of maybe do a sort of secondary comparison. But the IVR score is the primary outcome that we're using.
So have you and Mike designed it so that you're based on the IVR, the primary, you're either going to be able to show they're equivalent or that one is significantly better than the other? And how many more points on the IVR as a group does it have to be a winner?
Yeah, so it's non-inferiority. So it's powered. So we need 60 patients to show that lamotrigine is as good as mexiletine.
Oh.
And so basically to show that they're the same. And if it's better, then that's great. But the trial is designed to show that it's as good.
Well, that's great. And if you're trying to get that many patients, I'm sure the $10,000 help, but I suspect that you've had to find other resources from Dr. Hannah and others to pull this off. So,
we have. Yeah, I mean, because of I've got PhD funding as well. So
okay.
It has.
Congratulations.
Thank you. Thanks very much.
Next year, I guess you'll be done next year, right? So we'll have to have you given the final results.
Yeah, hopefully. I'm not sure if we'll have all the analysis done by MSG next year, but yeah, hopefully all the recruitment done. Yeah.
I think you're right, Rick. If we carry on doing the Shark Tank, we probably will have several feedback talks, won't we, as well as the main event.
Yeah, because I think they'll all be contribute to a small part of the bigger picture.
Yeah.
Now, another thing, James, we should keep track of the ones that didn't win. And if they still were able to find resources to do their project, we would want them to present. We need to think about that too when we plan the meeting.
Yeah, we could do a quick survey. That's a good idea. Great. I think I hand over now to Melissa, who's going to chair the next session.
Melissa, introduce yourself. Not everyone knows you're from Utah and you're a DPT, but just tell everyone who you are.
I think you did a pretty good job, but I'll follow it up.
Add a little more.
I'm Melissa McIntyre. I'm a PT at the University of Utah in the neuromuscular department there. I am also working on my PhD, so it's really great to hear all these projects and see what everybody else is working on. And so it's been a great meeting so far, and I really appreciate all the talks that have come before. With that, I'd like to introduce our next speaker, which is Leo Wong from the University of Washington, and he's going to be giving a talk on adapting MRI as a clinical outcome measure for FSHD, for a FSHD trial of prednisone and tacrolimus. Take it away.
Thank you so much. I want to thank the Muscle Study Group for inviting me to share our work. We're talking about FSHD and using some of the work that we've done in a clinical trial as an outcome measure. My financial disclosures are consultant for Mitsubishi Tanabe, Roche, and Fulcrum Therapeutics, but nothing that I'll talk about involves them, and so it should be safe. Just really quickly, FSHD is a disease that presents with facial proximal arm weakness, with wing scapula, followed by weakness of the foot dorsiflexion and the hip girdle muscles. And inflammation can be seen on histopathology. And our work really came out of the work of the Seattle Wellstone Project. And that is a collaboration between University of Washington and University of Rochester. I know that Rabit Will is on, and this was in close collaboration with him. We followed 32 patients longitudinally over a year, and we looked at STIR MRI, quantitative T1 MRI, functional outcome measures, muscle, mRNA-seq, and muscle histopathology. So what we found on muscle biopsy is that the STIR-positive muscles had higher expression of the DUX4 targeted genes, as well as genes associated with inflammation. So that was what we were targeting, inflammation. We also saw that over a year, there was a small change in overall T1 fatty infiltration of all the patients. So our hypothesis at that point was that normal muscle was T1 dark and STIR dark. There was this transition state that possibly represented edema, but to a neuromuscle neurologist, that wasn't really clear what that meant, or inflammation. And that is a set of muscles that were T1 dark and STIR bright, STIR positive. And then after this transition state where there's inflammation, where there's damage, it then gets changed into fat replacement, T1 bright, and it could be STIR bright or STIR negative. And so here's a little bit more of an example. Here is the edema I'm sorry, here's the active change in STIR1 hyperintensity, and you can see it here as well as here. And then we think that this could be a transition state, and then it transitions into a T1 bright muscle right here, and these muscles tend to be weaker. So what does this represent on muscle biopsy? This is what we found, was that STIR positive muscles, a larger percentage of those muscles represented inflammation or active myopathy. If you were STIR negative, you only had active myopathy or inflammation about 23% of the muscle biopsies that we looked at. So we looked at one of these patients in which we saw that they were STIR positive and also that they had inflammation. And so this was the biopsy from one of the patients in the cohort of the 32 patients. And you can see that there is variation in muscle size as well as inflammation that centered around the vasculature. So the question that we had was, for a patient with this muscle biopsy, with this inflammation, with STIR brightness on the MRI, if we treated this inflammation with tacrolimus and prednisone, would we be able to reverse the inflammation and reverse the STIR brightness? And that was what we were trying to do. So the patient that we were able to put in a clinical trial was one of the 32 patients in our Wellsome project. And it was a patient who was 45 years old. He had a genetic pathology of 9 D4Z4 repeats. He didn't have any facial limb weakness, but had problems doing a deep knee bend and walking on his toes or heels. So definitely some weakness. This is the clinical trial that we enrolled him in. It was a 12-week clinical trial akin to treating inflammatory myopathies. Was a 3-month trial, empiric trial of immunosuppressive treatments. And so what we had was we treated the patient with prednisone first 4 weeks at 40 milligrams per day, the next 4 weeks is 20 milligrams per day, and then a quick taper down to the last 4 weeks not having any prednisone. The tacrolimus that we used was 2.5 to 3 milligrams twice per day, with a trough level that we were aiming for at 4 nanograms per deciliter. We did functional outcome measures, strength measurements, and muscle MRI measurements at the beginning of the trial and at the end of the trial. This is what we found on the MRI. This is looking at STIR images. This is just a really cool stitching together that our collaborators did. Here, the bone that you can see here is in green. The fat, T1 brightness, is in yellow. The muscle, that's mainly the voxels, mainly representing muscles, are red, and the STIR positivity here are blue. And you have to go and stitch out some of the fat as well as the blood vessels. Here on the left is the baseline, and on the right here is at 12 weeks. There wasn't much change, and I have a little bit more quantification data in the next few slides. This is what we saw on a T1 looking at fat fraction. This is through the legs here, and you can see that there's some asymmetry between the right and the left. There's also some infiltration of the muscle through the T1, and you can see that it's pretty stable over the 12 weeks. So when we quantified it, we saw that at baseline, the STIR positivity was about 2% of the muscle that we could measure, was 3% at 12 weeks, and was the fat fraction, it remained about stable at 20%, both at the baseline and at 12 weeks. We also did a functional outcome measure. It was a 6-minute walk test, and it was really remained unchanged at baseline, being 600 meters, and also at 12 weeks was a decrease of 4 meters that probably isn't statistically or clinically significant. So our conclusion was, one, tacrolimus and prednisone did not reverse the STIR hyperintensity or improve the strength or functional outcome measure, and probably was not a treatment in which the benefits outweigh the side effects for the treatment of such a disease. We did find that T1 fat fraction could be an outcome measure for clinical trial, and that has been borne true in clinical trials that are currently going on with the Fulcrum Therapeutics. And it still remains to be found out in terms of how we can use STIR positivity as a measure of reversing active myopathies or immunoactive muscles. With that, I want to thank my collaborators through the University of Washington, Seattle Children's, and Fred Hutch. And finally, really want to thank our funders, and without their help, we wouldn't be able to do this study. And this is the Friends of FSH Research. So I really want to thank them. I want to thank you and the over 120 people who are listening on a Sunday morning, probably not going to church. And thank you for your time.
Thank you, Dr. Wong. We had a question crop up in the chat by David Johnson. He asked, did you hit our goal tack through consistently? How frequently did you check?
Right, so we think we did hit our trough levels. We checked them about once a week for the first 4 weeks, and then decreased it over the next 8 weeks to every 2 weeks to then every month.
Great, thank you. I think we have about a minute left or so. If anybody has any additional questions. As a physical therapist, I really liked having the functional outcome measure there as well. Because I think sometimes when we look at like MRI and stuff like that, linking that to function is always the first question that crops up in my head. So I was very happy to see that on there. Okay, thank you very much for your talk. Okay, next up we have Dr. Solomon Bai of University of Texas Southwestern. He will be talking on racial disparities in skin tone representation of dermatomyositis rashes.
Excellent, thanks so much. Let me share my screen here and get the PowerPoint pulled up. Can you see that okay? All right. Yeah, sure. Let's try that again. How's that? Oh. Sure. Yeah. How's that? Excellent, great. So thanks for the invitation to speak. It's an honor to speak to all the MSGers here and share what we've been doing with work regarding racial disparities in skin tone and how that's represented in medical education literature for dermatomyositis rashes. As part of the Exercise Institute that I work for, I think I'm contractually obligated to give a plug for exercise. We're deep into this great conference, so perhaps stand up, finish strong, do some squats, and the conference is almost done. We'll talk about racial disparities here, and in this context, these are just my disclosures. They're irrelevant to this study. And so I want to put this into context for you in the relevance and how it affects our patient-physician relationship here, how we got to the data that we got to, and then future directions. By the end of this talk, I hope you have an appreciation of what work needs to be done to help change the field. So the background related to this is that we know in dermatology that skin tone representation disparity exists. One survey took a look at dermatologists who had just finished their training, and about half felt they were not equipped to diagnose diseases in skin of color. And when you take a look at dermatology with different methodologies of whether it's journal articles, medical education literature, where the representation of different kinds of skin tones land, there's a decreased proportion of darker skin tones. That's across different areas within dermatology. The exception was with sexually transmitted infections, which had a higher proportion in darker skin tone representation, which doesn't match the clinical picture, what we see in terms of incidence and prevalence. And a recent example of this is with COVID-19 skin manifestations. Several studies came out also taking a look at how often darker skin tones are represented, and it's also disproportionately low. There's a patient-led initiative called Brown Skin Matters that's trying to take this into the patient's hands and curate a library of image that gives a better representation of what different rashes look like with different skin tones. And I think the importance, before we jump into the data, is fourfold. One is implicit bias. There's misrepresentation of the disease state. And so if you're not educated on what it should be looking like, you may be basing it off of stereotypes or simply miss it. You might harm the patient-physician relationship. Education early on from med school to residency, fellowship, and continuing education, you may have a distorted view of what these rashes should look like in certain populations, or you just may miss them. And that affects clinical care with delayed diagnosis and opportunities for treatment. And lastly, on clinical trials, we know with the cutaneous dermato disease area severity index, we rely upon what we're seeing on the skin. And so these are important factors that we should be cognizant of as we treat these patients. So we wanted to keep this study relevant to today's learner. And so the method that we used was we took a look at online resources and textbooks. So within textbooks, we searched for the different fields that may encounter some of these patients: derm, rheum, neuromuscular, neurology, internal medicine, and then myositis as a general search term. We limited it to the past 10 years, and it used the most recent edition. It was based off the UT Southwestern Library, so online resources only. Just keeping in mind the learner today on the wards or in the classroom, quickly taking a look at their online resources and consulting to understand what disease they're trying to learn about. The image databases we used were UpToDate, Visual DX, and DermNet NZ. The inclusion and exclusion criteria were relatively simple. We wanted a color image of a dermatorash. Non-rash skin that was visible was important because that can obscure a grading of the skin tone. And then the exclusions build off that. So if we couldn't tell what the skin looked like, if it was black and white, if there's poor lighting, if the rash was unclear, or we thought it was unrelated to DM, we would exclude it. And the method we used was looking at the Martin-Massey scale of skin tone here, and I put it on the right. And it's used as a social science grade of what skin tone looks like. In dermatology, we know that there's the Fitzpatrick scale. That was initially developed to see how white skin tones respond to UV light, and there were 4 classifications of that. Later, 5 and 6 were added to it for darker skin tones. So we felt that wasn't the most appropriate and skewed in one direction, which is why we chose this scale. It's used in high-profile social science studies. And there's several scales that could be used, but we thought this was the most appropriate one. And 3 authors of the paper independently graded skin tone, and we put it within one of these categories for MMSCS score. We binned it with 1 to 2, 3 to 4, 5 to 7, and 8 to 10, a priori thinking that most of these images were to land between 1 and 4. Images with large discrepancies were excluded. And what we mean by that is if 2 authors chose an image that was a score of 1, and another author chose that same image to be a score of 5, you skipped a bin. That was a rare occurrence, but we just set that rule a priori in case that the image was unclear. And then we ignored the caption to exclude other bias that might exist. So the results of this, we took a look at 93 textbooks and 3 online databases to give us a total of 561 analyzed images. And you can see that the distribution clearly leans towards dermatology textbooks, which makes sense. And then we have our subspecialty textbooks there as well that we took a look at. And you can see that the distribution of skin tones is heavily skewed towards 1 and 2, so the lighter skin tones. And then the darker skin tones, 5 through 10, make up less than 15% of the total images within these resources. And the important data points here is that, we started with 621, we excluded them because of the poor lighting, black and white, whatnot. 563 were analyzed, and only two were excluded because of large discrepancies. What we found is that there is no significant difference between the scores of the textbooks and online resources. The numbers started to get too low when we went to the darker skin tones to have high confidence in terms of the significance of the data. But you can see from the graph on the right that the darker skin tones are primarily represented in dermatology textbooks. And that's what we saw with the data there. You know, rheumatologists and neuromuscular specialists see these patients most frequently, and only 6% of those textbooks had skin tones of darker color. And then even within the different fields, again, the numbers were too low, but we're not finding a difference between dermatology and other subspecialties with the darker skin tone. And in terms of the inter-rater reliability, it was quite high. And again, only 2 images were excluded based off our previous criteria. Importantly, I think online databases with the brown skin matters, we didn't analyze that. They don't have any myositis pictures, but taking a look at up-to-date Visual DX and DermNet NZ, it's a great resource for finding what these pictures and these rashes look like. We have a fantastic teaching PowerPoint now with all these images, but they could do better in terms of their representation. So future directions, one is to take a look at medical journals and representation there. The second is looking at socioeconomic status and diagnosis, treatment outcomes as that affects for dermatomyositis and how these patients do. And then qualitative patient interviews to understand how they perceive this bias and if they have any changes to their care that they would like. Special thanks to Lisa Christopher Sine. She's a rheumatologist, director of the Myositis Center at Hopkins. Colin, Sophia, students who helped with this project, Dr. Salter, biostatistician who helped us, the UT Southwestern Neuromuscular crew, a fantastic group, myositis support and understanding, several mentors and inspirers along the way listed here. Many of them are present today. And then, UT Southwestern is undergoing renovations. And these pictures were going to be filed away somewhere. So I took a picture and I think you'll see some familiar faces there. Happy to take any questions.
Boy, that picture is scary. And I don't see why Wilson Bryan, I don't know why he wasn't in there. He must have skipped out on the picture. But thanks for, when you say they were going to do some rearrangement, that I guess means trash cans. So thanks for salvaging them. And I thought this was a brilliant idea. When I read this abstract, I just thought it was very, very clever. So thanks for putting this together.
Oh, thank you. I think there's lots of room to expand this in several of our diseases.
You might be able to get it published in a really, upper echelon journal, but if you can't, this would be great for the RNMF journal with some pictures. So let me know.
Yeah, great talk. We did have a couple questions roll through in the chat. The first one was, the main source of clinical images for rheumatology is the ARC image bank, which is online. In their experience, this is pretty equal representation in terms of skin pathology. Was that reference included?
That's a great question. We did not include that when we were searching through different databases, but that's something that we could look to include and get some data from there as well.
Okay, great, thank you. And then we had another question. Is there any data on delay of diagnosis in patients of color with dermatomyositis?
Thanks, Eric. Great question. That's something that we're looking into. It's a little hard to define. As you know from the medical records, patients show up with all sorts of different ICD codes and documentation is not great. They maybe got a biopsy, maybe elevated CK, and the diagnosis is still unclear. But I'm taking a look at Parkland versus UT Southwestern, where we have distinct populations, to take a look at, when was the first CK that was known to be elevated? When was the diagnosis made? When did the CK normalize? And what was their total steroid dose? And hopefully that gives us an insight into, was there a delay? How are they treated? And was there a treatment outcome change?
Perfect. Thank you very much. So I think the time is up for our next talk. It's my pleasure to introduce Stacy Sullivan from Mass General Hospital. She's a speech therapist, and she'll be giving a talk on longitudinal dysphagia assessment in patients with cystinosis using the modified barium swallow impairment profile. Take it away, Stacy.
Hello, thank you so much for the introduction and opportunity to share updates on this work. Just a little note is that this work is funded by the Cystinosis Research Foundation. And can you guys see my slides? Wow, that is so helpful. Thank you very much. Okay, so just as a reminder, nephropathic cystinosis is a rare lysosomal storage disorder. These patients present with, among many things, distal greater than proximal muscle weakness, prominent swallowing and breathing difficulties. The exact pathophysiology of the muscle impairment is not clearly defined, and there are many questions regarding the adaptive capacity of the muscle tissue. So an opportunity to work with these patients and explore potential treatment targets that can benefit function and quality of life. So this condition has some similarities to inclusion body myositis, such as hand weakness and difficulty swallowing. So I did have an opportunity to speak about this study a little bit last year with this group. And as an overview, we had phase 1, we brought 20 patients in for a clinical trial readiness study, again, supported by CRF. And we did some baseline measures, including comprehensive neurological testing, video fluoroscopic swallowing assessments, and some breathing testing, including maximum expiratory pressures and peak cough flow. Patients came back 1 year later, and we repeated the testing and introduced a 5-week exercise training program called expiratory muscle strength training with the trainer device called EMST 150. These patients trained on this device for 5 weeks. We decided to select this as a potential treatment target. There's literature in other disease populations that suggests that this training can improve both cough, force, and function, as well as improve function of the supraglottic swallowing structures, and therefore improve ability to swallow and mitigate aspiration. So after the first phase, we also included patient-reported outcomes. We noted that a high number of patients were reporting dysphagia symptoms, and less patients were demonstrating these dysphagia signs on the way that we were evaluating them. We were using, performing the video fluoroscopy and then using an 8-point ordinal scale called the penetration aspiration scale, which basically describes the airway invasion for patients. So level at which contrast enters the airway and then the patient's response. So using this kind of crude metric, we were not able to really capture the severity of dysphagia that these patients were reporting. And this was significantly important to us because there were a lot of quality of life measures in these surveys that we were using, and so the patients were really struggling, but we weren't capturing exactly why. So after the respiratory training session, the patients came back, and we did demonstrate some changes in maximum expiratory pressures and peak cough flow, which were consistent with findings in other bodies of literature working with other disease populations. However, there were some not statistically significant changes in improvement in swallowing. So we again felt like this training may benefit this patient population in terms of breathing, but we weren't able to really move the needle on anything related to swallowing. At that point, we took a look at a different way to evaluate swallowing. The Modified Barium Swallow Impairment Profile is an evidence-based standardized assessment tool which breaks down swallowing physiology into 17 components. And the rater goes through and rates each component individually with a scale of 0 to 4, with 0 being normal and 4 being most impairment. So we took this data that we had collected through the course of phase 1, 2, A, and B, and launched a retrospective analysis to see if we could better understand these patients swallowing and hone in on more specific treatment targets. So because of COVID, we were stalling a little bit with some recruitment for phase 3 anyways. So it was a great time to take a look at data we already had. This proved to be very interesting, and we did see that we were able to isolate more specific impairments, specifically lip closure, bolus control, tongue function, essentially, laryngeal closure, tongue-based retraction, and pharyngeal stripping. These findings much more significantly correlate with patient report of difficulty swallowing and allow us to think of what to do about it in a different way. So in summary, this advanced analysis was extremely useful in identifying changes in the oral stage of swallowing. Oh, I think I skipped a slide. Yep, here. So just to add on the MBS IMP, there is the ability to create total impairment scores. And we do see here that when we separate the oral and pharyngeal phase of swallowing, the oral score findings did detect slight trend in decline of function over time, and the same with the pharyngeal total score. So that was interesting as well, to be able to capture these really slight changes as we look at these patients longitudinally that we hadn't been able to describe before either. So this is a really interesting tool to use with this patient population, both to better select treatment targets, which aim to help the patients swallow and improve quality of life, and then also measure discrete changes over time. So we will use this moving forward, and in addition to looking at some other things related to these patients and how they're affected by myopathy. So thank you so much to the amazing team and everyone that's come together, and of course the Mass General and the Cystinosis Research Foundation, because we've been able to do some really cool work. So happy to take some questions.
Very nice talk, Stacey. I really appreciate it. I think swallowing is something that we're always kind of seeking on how to measure functionally, and so that was a really nice talk into it. Would like to open up the message board, or anybody. Oh, here we go. Kira Berggren, sorry, Kira. Did you see any changes in esophageal functioning?
So we did not look at the esophagus, and part of that was our initial intent to measure swallowing just using the PAS, and then we were doing the IMP retrospectively. The IMP does look at esophageal clearance, at least, and we didn't have enough to consistently talk about it because we weren't looking for it initially, but absolutely including that moving forward.
Great, thank you. Kira says thanks. And does anyone else have any questions? Okay. All right, great. Thank you so much, Stacey. We will move on to our next talk. So our next talk will be CNT COVID survey by Dr. Ricardo Zuccarino from NEMO. So go ahead and take it away.
Okay, thank you for inviting me. Just take, okay. Okay, here we go. Do you see my screen? Okay. With our study, we just want to evaluate how much COVID-19 impacted in CMT population. And for doing that, just we organize a simple questionnaire for CMT patients. And we asked them just to answer the questionnaire if they had CMT and they got COVID-19. with the support of the Italian CMT Association of Patients, CMT Rete, and in collaboration with the INC, we just distributed the survey by email, and we just reach out all members in the contact registry in the US and in Italy through the Italian CMT registry and by the HMT Rete members newsletters and other social networks. The questionnaire was just designed answer to patients specific question about their symptoms, the presence of pre-existing risk factors, the presence of sleep apnea, and eventually, if they had it, the presence of non-invasive ventilation, and if there was an hospitalization, if they were admitted to the ICU, and if there was an hospitalization, if a ventilator was required during the time in the hospital. This is the survey, and you can see what I have already told about the different symptoms and the different risk of factors. The results just show a lot of answer from patients, but not very much anyway, because a total of 153 patients completed the survey in a pretty much long time, because we just send out the questionnaire between May 2020 until January 2021. And we can see the demographics of the different distribution of different types of CMT who answered the questionnaire. And this is the different symptoms and the risk factors the patients had before got eventually the COVID-19. And of course, our study has some limitation, because in any case, our data comes from individuals who we have not examined before, and they just ask them to answer this online survey. And another point for us a little bit tricky but important is that we ask them only to answer if they have CMT and got COVID-19. And the fact that many completed the survey but did not have COVID-19 raises some question to us about the actual percentage of patients with CMT and COVID-19. And we consider that is much less than 13% of CMT population cited here. And we are aware that we received just 100 of answer, and usually the INC registry for CMT included more than 1,000 of patients. But again, we ask just only if they got COVID. Our conclusion at the end are that COVID-19 is not common in CMT patient population, and whether there are increased risk for patients with rarer forms of CMT with significant respiratory compromise remains unanswered, and we require investigation focusing exactly on these subgroups. Thank you.
Thank you very much for that talk. Really nice and informative. Tara asked in the chat, do you have any data on patients with CMT and side effects from the vaccine?
No, unfortunately, because our survey was just about COVID-19 and before, sometimes in the vaccination. So, and we just didn't collect any information about eventually side effect from the vaccine. But from our clinical experience, we can say that are the same of the other population. There is not any different for the normal population.
Okay, perfect. And then we had another question asking if your study included children.
Good question. The age was from 18 to 90. So we didn't include children.
Okay. Very good. Yeah, very nice. And then you may have mentioned it and I could have missed it, but how did you administer the survey? Was it through your clinical population or just kind of a survey out through local foundations or something like that?
Yeah, was just send out using the INC contact registry online and by the support of the US CMTA Association and another Italian association, the name is ACM Tirete, who reach out, all patients are included in their registry.
Okay. Perfect, thank you. And then Tara asked again, was the goal to survey subjects when they currently had COVID, or was it a retrospective, or was there a retrospective component?
We just wanted to make a picture of what happened for CMT population and COVID, in particular, how many patients got COVID-19 and how much eventually the virus impact the life of these patients. This was just the goal to understand if there is an increased risk for CMT patients.
Okay. Perfect. Well, thank you very much. If there's no further questions, or wait, hold on, actually.
Thank you.
I think we had one more, if you're willing to stay on, and we have time to ask for it. They asked, did you look at the interruption in therapies and other services as well as clinical services? Nope. I think we stepped away.
I'm here, sorry.
Gotta love Zoom.
Sorry, I just miss a part of your question eventually.
Okay, I will repeat it. The person is asking, did you look at interruption in therapies and other services as well as clinical services?
No, we were a little more interested eventually in understanding how many patients of CMT who got eventually COVID-19 worked in the hospital, just to understand if eventually this part of population had more was easier for them, just got COVID-19, but we didn't find anything. And we just look in the clinical features, and we didn't investigate other field, for example, like therapies or other services or clinical services.
Okay, perfect. Thank you so much. I think our time's up for the next talk, but I will say that Tara made a comment in the chat of areas you could look for more data. So that might be helpful to take a look at. So moving on, we have Timothy Walton. And sorry, I just lost my space. He's going to present on a case series of myasthenia gravis patients prescribed subcutaneous immunoglobulin therapy and monitored by patient-reported outcome measures by specialty infusion pharmacy using Solmetrics. Take it away, Timothy.
Okay, let me share my screen. All right. Can you guys see my slide deck?
Yeah.
Is that right there? Okay. There, can you see it now? Okay, that's it. Okay. Gotcha. All right. All right. Do you see the? Oh, okay. Apologize for that. All right. So let me go back to. I'll share my other screen. Hang on a second. Okay. I have three monitors. I apologize for that. Okay. All right. Good afternoon, everybody. My name is Tim Walton. I am the Director of Data Quality, Health Outcomes, and Research here at Solio Health. And I'm coming live from Kansas City. Go Chiefs today. Hope they win. And I'll be looking at a case series of MG patients getting sub-Q being monitored by PROMs through a specialty pharmacy. Right, go to the next slide here. All right. So all of us on the call know that documented use of sub-Q in MG patients being treated and monitored by PROMs through a specialty pharmacy particularly is extremely limited. We've presented here a case series of 13 MG patients who were monitored through Solio Health. And what we looked at were patients who were on therapy from October of last year until August of this year. We looked at patient history, dispensing records, MG symptoms, ADRs. We looked at the MG Qual 15, the revised one, the MG ADL. We looked at pain scores, either related or unrelated, depending on how the patient responded to our surveys. We looked at fatigue scores. We used the MFSS or the modified Roche-built fatigue severity scale. We also looked at ataxia, sensory perception, weakness, and we have an internal quality of life measurement we use called the Solio Wellness, as well as we looked at the type of payer and the type of physician referral source. Looking at our demographics and dosing information, we have 113 patient visits during this time frame, and that is a mean average of 9 per patient. And the way that we capture this data is that when the pharmacist calls a patient on the telephone and does a dispense, they have the patient answer a series of questions related specifically to their disease. And also, if the nurse goes out to the home setting, when they try to teach the patient how to do sub-Q administration, they also ask them questions. The population that we looked at, the mean age of patients is 50. You can see the range in these slides for each of these headers. 92% were female. The average height is 65 inches. Our weight mean is 85 kilograms. The mean ideal weight of these patients is actually 57.92, and their mean BMI is 31.1. What is of interest is that for the type of payer for sub-Q, 70% is government paid, 23% is commercial, and 7% is PBM. Or if you look at the cell right next to it, 9 patients were government, 3 were commercial, and 1 was PBM. Physician referral sources, we had 8 academic physicians compared to 5 non-academic physicians. As far as the dosing information, we had a mean average of 28 grams over an average of 5 sites, and you can see the range is 1 through 7. And the frequency, it was 7 days with one patient who had a 14-day frequency. So this is the outcome measures of the 13 patients from baseline of when they first started having sub-Q administered to the last documented case. And so I know this is kind of a lot to look at, but the first rows, we have MG-ADL, or the MG-QoL-15, the MG-ADL, pain, fatigue, our wellness scores, and the number of assessments performed on the patient, as well as if they're currently on sub-Q. I highlighted the boxes where you show where there was a change in an improvement in these outcome measures. And now you can see the asterisks are patients who decided that they opted out of collecting certain outcome measures, whereas you can tell there are some patients who kind of stayed static through their therapy, and some actually got a little worse. But like I said, 12 out of 13 are currently still on sub-Q, with one who was switched to IV because the physician felt that the sub-Q wasn't helping their current condition, so they wanted to try IVIG. So the average number of PROMs per patient, we have 72 QoL-15s, and I need to preface some of these numbers because some patients opted out, like we saw on the previous slide. So of the 113, we have 72 QoL-15s, which is about 7 per patient, MG-ADLs with 7 per patient, and you can see the rest here. And all 13 patients improved through their treatment. Their ataxia, sensory perception, and weakness improved as well. And we have specific questions that we ask patients in each specific section. Looking at the ADRs reported for the 113 infusions, we see that over almost 46% reported no adverse reactions at all. However, 17% reported headaches, and then redness and swelling infusion site, followed by migraines, myalgia, dizziness, fatigue, nausea, and rash. So the internal ciliary wellness scale that we use here, we specifically ask questions on their energy levels, what their mood is, what their sleep patterns are, what their stress levels are, and how they're feeling during their health and wellness. And we're actually currently trying to validate this survey and actually analyze what the data looks like pre-COVID and what it looks like during all of 2020 and how it looks now that people have been vaccinated. There's other treatments out there. So we're going to try to look at all that data together and see if there's any actual change in these particular questions and correlate them to the problems that we've discussed previously. So SoulMetrics allows the frequent data collection of information by trained clinicians, either the nurse in the home or the pharmacist over the phone, not readily available to physicians like yourselves between routine office visits and then sub-Q self-administration. Pharmacists will also be able to make recommendations related to side effect management and promoting better outcomes and consult with treating physicians on the course of appropriate clinical action. And then by reviewing SoulMetrics data, physicians then are able to optimize therapy and clinical outcomes in patients with myasthenia gravis. And the MG patients we looked at, there was a representation of both myasthenia with and without acute exacerbation. So I just wanted that we didn't just pick one over the other. We looked at both types of patients. And I'm done.
Great. Thank you, Timothy.
Okay.
We have about 5 minutes for chat. There's been a lot of chat going on. And this might be a good one to maybe have people come on over the mics and ask their questions. Okay. I think, Tashin, you were one of the first to come up. If you'd like to kind of bring up some of your thoughts.
Sure. So, Timothy, it looks like a bunch of assessments were done with no real relevance to myasthenia. So myasthenia, for instance, is not a disorder that is known for sensory abnormalities or ataxia. For some reason, these were collected, even though that's not typical for myasthenia. The other challenge is, which is something, and a number of home infusion companies have explored this in the past, which is to do these assessments at home. But the challenge lies with the fact that the evaluators who are doing this, which is the nurses or the pharmacists, are employed by for-profit companies like Solio, which means that their assessments can potentially be biased towards showing a positive outcome of the treatment rather than with that. So in the past, when we have been approached to do something like this project like this, we had insisted that the assessments need to be done by the treating physician rather than employees of the company that is doing the infusion. But we were just surprised, and I think a number of the other chat questions are related to the scale of the outcome measures that had very little, the number of outcome measures that were done without any significant relevance to the disease. Sorry.
Okay. We consulted KOLs who treat myasthenia, and these were the ones that came up, but I can discuss that again with the team here.
Yeah, I would love to see the data on which the sensory ataxia or ataxia or pain or sensations were chosen.
Okay. I'll make note of that.
Okay, great. Thank you so much. There was another question that asked, could you give some detail on how patient-reported outcome measures and adverse event assessments were done?
Right. So we have a collection of surveys that we collect on various diseases here at the company, and when like I said earlier, when the pharmacist calls the patient, there's a certain list of questions that we ask them, and they could be anything that Joint Commission requires, or things we require, or things that payers or manufacturers or specific physicians require for their patient populations. And that's how we collect them on these assessments. And so that's how we then document those on that slide that you saw, is that these are all patient reported over the telephone or in person with a nurse who, like I said, who was there, training and then monitoring post administration in the patient's home.
Great, thank you. And in that same vein, how was muscle strength examined? Was asked by James.
Let me see. Well, we just administer the questionnaires, so that's all. That's all we do. We don't do the MGC in the home, just for obvious reasons. And so we just ask patients questions.
We have about a minute. Does anyone have any additional questions? I think we got generally most of the ones in the chat, but might be good to kind of take a look back, Timothy, and respond to some of those. But if anybody has a quick verbal question they'd like to ask, we have about a minute for that. Okay, James just asked, I will end with this one. James just asked if the questionnaires you used have been validated.
Well, our internal one, not yet. As I mentioned, we're in the process of validating, but I would think we all know that the Qual 15 and the MG-ADL have been, and so is the MFSS one. So I'm not sure how to answer that one. Oh, the weakness one, those are internal questions that we use here at Solio. But I will take the discussion earlier, and I'll go back to the team here, and we can reassess that assessment going forward. So I appreciate the critique on that one. Thank you.
Thank you very much.
Okay.
All right. So next up we have Valdemir Lyarski. He's going to give a presentation on flow cytometry and sorting of single antibody screening cells from frozen muscle tissue. So take it away, Dr. Lyarski.
Yes, hi. Thank you so much for that invitation. Let me bring up my talk. That scene. Okay, thank you. So once again, I'm Vladimir Lyarski. Thank you so much for the organizers for this invitation. This is my first MSG meeting, but it's been actually very impressive. So I am a rheumatologist at the University of Chicago, and I'm happy to share this presentation on flow cytometry and our application of a new technique to sort single antibody secreting cells from cases of inflammatory myopathy. I have no disclosures relevant to this presentation. Basically, the goal of this project was to develop a method to isolate single antibody secreting cells, or ASCs, from frozen muscle biopsies of patients with IIM, or idiopathic inflammatory myopathy. This stemmed from our own interest in the immunology as well as comparison of IIM pathophysiology with other disorders in rheumatology, such as lupus nephritis and rheumatoid arthritis, and the role of humoral autoimmunity that is played in those. Obviously, this audience probably does not need this to be mentioned, but there are multiple hurdles to trying to do this analysis. One is that there's obviously very limited tissue availability since the disorders under study are rare. And even if you get fresh tissue, you actually do not necessarily know the diagnosis immediately, since other studies have to be done to confirm the diagnosis. And unfortunately, there's a discrepancy between cutting-edge techniques such as single-cell RNA-seq, but these are not really compatible with frozen or material that's been stored. So with this in mind, we enrolled patients who had pretreatment muscle biopsies that were read at the University of Chicago and fulfilled pathologic criteria for either dermatomyositis or inclusion body myositis, and I'm giving a reference for that criteria. And since ASCs are not normally resident in normal muscle, for our pilot work, we actually used the identified human tonsil for our initial experiments. Now, there is the Accelerated Medical Partnerships Protocol, or the AMP, which has, among other things, shown that you can use frozen kidney tissue and store it with digestion and flow cytometry and RNA-seq data obtained from those specimens, but that utilized a proprietary medium called CryoStore. And so we wanted to see if we can apply or obtain ASCs from biopsies for muscle, which, as has been mentioned, is prepared differently. It's prepared in liquid nitrogen cooled isopentane. So we took tonsil and we took paired samples, and we either used fresh tonsil or tested CryoStore or liquid nitrogen preparation with isopentane, and then digested the tissue to see what we could get. So this is the first figure. So panel A, you can see here on top, this is fresh tonsil, and this is flow cytometry performed to isolate single cells. And we then stained for CD19, denoting B cell populations, followed by selecting CD27 intermediate or high memory cell populations, which were IgG negative, and then looked at CD138 as our primary plasma cell stain. And so you can see that this compares very nicely to the data from the CryoStore frozen samples, as well as very similar to the isopentane frozen tonsil. So there was really, based on a qualitative look, not too much difference between the three preparations, which were so exciting. This is a quantitation here in B, comparing fresh versus frozen versus isopentane frozen samples in red. And this is for each parent population. So pretty much in every instance, the isopentane was non-inferior, and actually in some cases was slightly better than fresh, and certainly comparable to CryoStore. So once we got this data from tonsil, we then subjected the same thing to dermatomyositis and IBM, to the same pipeline. And so again, we could see nice cellular material that was isolated, excluding doublets and triplets. We added calcine to this, which we previously did not use in tonsil, just to ensure that we had a cell viability from these samples. And again, we subjected the same to B cell CD27 and IgD signatures, as well as CD138 staining. And this is just the three-group comparison to quantify this data. So we have dermatomyositis in white, IBM is in the black squares, and then tonsil is in triangles. And each of these represents a singular biopsy. And so we could see that we had a very nice recapitulation of live cells from both IIM cases, and there were 3 biopsies each, and this compared nicely to our tonsil experiments. We then wanted to make sure that we're actually able to get usable RNA, so we have cells, what look like cells on flow cytometry with calcine. The question is, do we have RNA? So we sorted these single cells into plates, and then we just subjected them to traditional PCR. And we have targets which are BLIMP1 or PRDM1, which is a plasma cell transcription factor, BCL6, which is B-cell transcription marker, and then we expressed Ig heavy and Ig light chain, immunoglobulin heavy and light chain, and then our control is beta-actin here. Negative control is illustrated by NC. So you can see we subjected 2 samples or 2 cells from IBM and 2 samples, and 1 from tonsil. And you can see here that we clearly have intact RNA and DNA from which the PCR was successful, confirming that this is a post-germinal center program. And we have just a comparison here of relative fold expression of PRDM1 and BCL6 across a number of cells, and we can again see that we have a post-germinal center development B-cell stage that would be predicted from the prior data. So in summary, we recovered similar proportions of CD19 positive and downstream ASC cell subsets by flow cytometry from tonsil, and the isopentane freezing was really non-inferior to cryostore. And following this digestion of 3 dermatomyositis and IBM biopsies showed similar proportions of sorted ASCs, and we confirmed expression of BLIMP1, BCL6, and immunoglobulin. So our approach really establishes the feasibility of obtaining live antibody-secreting cells from stored sample of frozen muscle tissue, really opening up the potential for downstream applications. And that's the end of my talk, and I'd like to thank all the members of my lab and my collaborator.
Okay, perfect. Thank you so much for the talk. First, we just had a comment stating, I worry that insurance companies will try to use data from specialty companies to guide therapy. I think these studies need to be carefully vetted. Do you have any thoughts or comments on that statement in the chat?
I'm sorry, this is to me?
Yes, sorry, or just kind of a general comment, and so I just didn't know. Oh wait, I apologize. I think this came potentially from the previous talk. So let's skip down to the next one. What was the average yield for immune cells from muscle cells for these experiments? And then a little bit of the description, if you'd like to look in the chat.
Oh, sure. So we had an excellent yield. So I mean, this data. So this data is submitted for review, and hopefully will be published possibly by the end of the year. We have plenty of cells for single-cell RNA-seq. So this is using half of a standard muscle biopsy, a surgical muscle biopsy. So this is not a core. This is an excisional surgical biopsy as is standardly done at our center. So cell death is obviously an issue. So we've done this. We can get live cells from biopsies as old as 6.5 years. So depending on what you want to use it for, we certainly have more than enough cells from even those old specimens to do just regular RT and single cell cloning of B-cell receptors. So that's easy. If you want to do RNA-seq, then you generally want to get some newer samples. So we've kind of validated and have everything working up to about 2 years. And we know that 6-year-old samples don't really give you good RNA-seq. But we have more than enough cells for RNA sequencing. At least we've used specifically the 10x genomics platform. So that would require about 10,000 cells and on the chip. And so generally we'd like to shoot for at least 100,000 cells isolated before we give it to the lab. So we've not had an issue there.
Perfect. And then we had another question on the viability of total cells across 3 conditions tested. Just wondering what that viability was.
Right, so that's a really good question. So viability varies. It can be as low as 10% to, so it really depends on 3 things. I mean, one was the adequacy of the sample before, meaning did it get to the pathline in time? Did it get frozen in time, or was it laying around for half the day, and have necrosed? And 2, obviously, is the age of the sample. So if you do like-to-like comparisons, such as we did with the tonsil, the viability actually was not shockingly bad. So offhand, I'd say it's about 60% live cell recovery from fresh tonsil. So about 40% of tonsil cells died, even when we collected the specimens. And so for the tonsil, for IP and CS, both were around 40% for the freezing condition. So the freezing actually did not impact a lot of cell death. And in fact, that kind of makes sense, because if you're familiar with the cryostore protocol, you have to put it for freezing in this medium for 24 hours. So it's kind of a slow DMSO-based freezing algorithm. Versus isopentane, you just check the tissue in and it freezes like in 10 seconds and you just have this desiccated piece of tissue. So if anything, I'm actually kind of, I'd say the IP is slightly better at minimizing cell death in terms of, you know that you're frozen right away versus waiting for 24 hours.
Okay, perfect. Thank you so much. I think our agenda, keeping on the time frame of our agenda, we'll move on to the next speaker. Okay, so next I'd like to introduce Aldrihara Alangari. Is a PT and PhD student at the University College London. She's going to be speaking on the effects of distal hereditary motor neuropathy on muscle structure, function, and gait patterns. This will be 2 case reports.
Yeah, thank you so much, Melissa, for the introduction. So I'll now share my screen. All right. Is it fine? Yeah, okay. So my name is Al-Johara. I am a physiotherapist and a PhD student at the UCL Queen Square Institute of Neurology. So DHMN in general is a rare inherited neuropathy. It's usually the clinical manifestation involve adult onset, slowly progressive, limb-dependent muscle weakness and wasting. It lays under the Charcot-Marie-Tooth disease or CMT umbrella. However, the sensory involvement and deformities are infrequent. With more than 10 types and more than 30 genes identified, only 20% of DHMN cases are genetically diagnosed. And mutation in HSPP1 found to be the most common. So today I present 2 DHMN cases from previous research at Queen Square compared to matched control. Measures compared are intramuscular fat semi-quantified using Mercury scale, isokinetic and isometric muscle strength measured by HUMAC, kinetics and kinematics of gait measured using 3D gait analysis. This case report was part of my training for the PhD study, which is aimed to provide preliminary longitudinal data using similar methodology to describe the natural history of DHMN and to investigate the effect of commonly used rehab intervention, including exercise and ankle-foot orthosis. So the first case is a 47-year-old male with no identified gene yet. Compared to control, dynamometry showed low torque values predominantly in plantar flexors, and MRI scans showed symmetrical calf posterior compartment involvement, and thighs had normal appearance. In comparison to healthy control, DHMN walked with a shorter stride length, and in pre-swing phase, vertical ground reaction force was less and was associated with less ankle power. In swing phase, knee power was greater, and that could be a compensatory strategy to help progressing the swinging leg forward. The video shows delay in heel raise in late stance associated with plantar flexors weakness. The second case is a 37-year-old male with mutation in BSCL2. Compared to control, dynamometry showed low torque values predominantly in dorsiflexion and in eversion. MRI scan showed asymmetrical calf lateral compartment involvement, and thighs had normal appearance. In comparison to healthy control, this case walked also with shorter stride length, and in pre-swing, ankle power was less. In swing phase, there was decrease in ankle dorsiflexion and dropped foot pattern. compensated by an increase in hip angle, hip moment, and knee power. In stance phase, knee flexion angle was greater, and vertical ground reaction force graph showed more force at the beginning of the stance phase, and that could be related to forefoot strike and dropped foot gait patterns. In previous studies, semi-quantitative MRI using T1 weighted images was helpful in identifying patterns of muscle involvement in cases with HSPP1. However, the quantitative MRI using Dixon techniques was more responsive over 1 year in CMT and had a strong correlation with clinical functional measures and hemodynamic. Studies in CMT described two different gait patterns, including plantar flexion failure and dropped foot, which was related to distal muscle weakness. And both patterns can be compensated with proximal involvement. So from the literature and the case studies, we can see that a variation existed between cases, and DHMN is not a homogeneous group of diseases. Also, the proposed methods have the potential to explore the relation between different muscle involvement pattern and gait in DHMN. Thank you.
Perfect. Thank you so much. What a wonderful talk. Does anybody have any questions? I'd like to open it up for questions now. Okay. Well, that was really enlightening. It's always, I think, really kind of a fascinating thing to look at how these diagnoses and resulting weakness can alter our gait mechanics and the compensations that patients do. And often these things are very difficult to visualize with our eyes. I mean, as PTs, we do it all the time and we try. And so giving a more quantitative and in-depth view of that, I think, is really interesting work going on in the field. So thank you for sharing that with us. If nobody has any additional comments or questions, we'll move on.
I guess I don't, yeah, this is Rick. I'm just trying to think. I really like that very detailed gait analysis too.
Yeah, thank you.
When you do these sort of gait analyses, this is a naive question. I mean, is there anything else you can do to ensure that the sensory is really completely normal? Because we're assuming that's what it is by definition.
Yeah.
It's all motor weakness, but you know, these HMN cases are, I know they have normal nerve conductions, but do they have any subtle sensory abnormalities? What do you think?
So with DHMN cases, they will be neurologically examined before being included in this study. So this is to make sure that their condition doesn't affect their sensation in general, and they also be examined if they have any other condition, for example, any.
Yeah. How about this? Because that was really not a good question, because it was sort of how about this? Can you tell the difference between, on this sort of analysis, between a CMT1A and a DHMN? Is there a difference in this type of analysis?
In gait analysis?
Yeah.
Well, so in terms of pattern of involvement, they can be very similar, especially with distal muscle weakness. So in DHMN, patterns of weakness observed in previous studies included anterior compartment or dorsiflexors involvement, and calf muscle involvement. These patterns were also observed in CMT. So it's actually, well, detriment, it originally lays under the CMT umbrella.
Yeah, right.
With its exclusiveness to the motor nerves. So in terms of gait analysis, no, I don't think we can differentiate between CMT and detriment using only gait analysis.
All right.
Yeah.
Well, that's fine. That makes sense. Thanks for doing this study on this rare group of patients.
Yeah, thank you so much.
Okay, next I'd like to introduce Dr. Xion Young Lee from the University of North Carolina at Chapel Hill. She will be giving us a talk on monotherapy with eculizumab in refractory acetylcholine receptor positive generalized myasthenia gravis.
Let me just share my screen really quick. Can you guys see my screen? All right, so my name is Xiaoyang Li. I'm one of the PGY4 residents from UNC. It was my great pleasure to be able to present here at the MSG meeting. So today I'm going to talk about monotherapy with eculizumab in refractory acetylcholine receptor positive generalized myasthenia gravis. So, as we know, eculizumab is a monoclonal antibody that targets the complement system. It has recently been introduced to treat, complement-mediated autoimmune neurological conditions such as myasthenia gravis and NMO. In 2017, the REGAIN study and its subsequent open-label extension showed that eculizumab is effective and well-tolerated in patients with acetylcholine receptor positive generalized myasthenia gravis, which eventually led to the FDA approval of this indication. However, in the trial, the eculizumab was mostly used as an add-on therapy to those refractory patients who are mostly already on other immunosuppressive therapy. So it will be interesting to know if we can manage myasthenia with eculizumab alone and to reduce the burden of chronic immunosuppression. So here we want to share our experience with two cases. The first case is a 32-year-old female who was diagnosed with generalized myasthenia gravis in 2010. She underwent a cystectomy a year after the diagnosis and was treated with mycophenolate from 2012 to 2014, during which time she had 3 exacerbations requiring hospitalization and plasma exchange. We decided to switch her from the mycophenolate to IVIG in 2014, partially because she wanted to become pregnant and there was some concern about teratogenicity. However, her symptoms were never fully controlled on IVIG, and she also had pretty bad headache with the IVIG infusion. So at that point, we decided to start eculizumab. As you can see from the graph here, this is the change in the quality of life for MG metric scores along with the initiation of the therapy. So as you can see here, the baseline score were pretty high. The quality of life, ADL, manual muscle testing, and MG composite scores are pretty high while on IVIG at baseline. And then here, the score dropped significantly following each induction dose of eculizumab. And the score had remained low for more than a year now since the initiation of the therapy. The mild increase in the quality of life and ADL score here in August was mainly due to the shortness of breath in her late pregnancy. At that point, she was 32 weeks pregnant, and then she actually just delivered a healthy baby about 2 weeks ago. So moving on to the case number 2, this one is a little bit more complicated. This is a 43-year-old female with history of lupus, mainly cutaneous lupus, and also recurrent ear infection, who was diagnosed with myasthenia back in 2011. She was initially treated with methotrexate monotherapy from 2011 to 2015 because of this coexisting lupus condition. However, she had 2 exacerbations requiring hospitalization and IVIG in 2015. And then at the time of discharge, she was on mycophenolate, methotrexate, and also monthly IVIG for maintenance, but she still had another exacerbation in 2016. So she underwent a thymectomy in 2017, but continued to have pretty severe myasthenia symptom and ended up having 2 exacerbations in 2018 requiring intubation. So at that point, we decided to start eculizumab. As you can see on the graph here, prior to the initiation of the eculizumab, she was on 3 immunotherapies, but still quite symptomatic in terms of the MG metrics. We decided to switch her from IVIG to plasma exchange because there's some concern about IVIG having interaction with eculizumab and reducing its serum concentration. So literally for the first 5 weeks, she will have plasma exchange in the morning, followed by the eculizumab loading in the afternoon, and thereafter every 2 weeks plasma exchange, followed by the eculizumab infusion. Her symptom improved quite a bit, so we're able to space out the plasma exchange to every 4 weeks. But unfortunately, there was a bump in the road. She had an infection of her cochlear implants, leading to an exacerbation, and end up in the hospital for a 5 session of plasma exchange. So after discharge, we have to do the whole thing all over again, reload her with eculizumab along with the weekly Plex. And her symptom actually settled down pretty quickly. So we're able to space out the plasma exchange to, again, every 4 weeks. And since then, she has been doing very well in terms of, myasthenia and also lupus. So we're actually able to stop the plasma exchange altogether in March 2020, and her rheumatologist actually stopped the methotrexate 3 months after. And since then, she has been doing really well on eculizumab monotherapy for about a year now. So as clinicians, we're oftentimes amazed by how well patients are doing with eculizumab, but I guess so far we still don't have enough data to support its use as monotherapy. In this case area from University of Missouri, dose reduction of the concomitant immunosuppressants such as prednisone has been reported after the initiation of the eculizumab, and 2 patients actually stopped the concomitant immunosuppressants by themselves and ended up on the eculizumab monotherapy, 1 for 2 months and 1 for a no period of time. And interestingly, learning from the experience of the sister disease, NMO, eculizumab monotherapy has recently been shown to be pretty effective and well tolerated in a subgroup analysis of a 33 NMOSD patient from the PREVENT trial and its open label extension. So it will be very interesting to see if we will have more data in that regard from our MG cohort. So to conclude, we report 2 patients with refractory myasthenia gravis that were successfully transitioned from multiple immunosuppressants to eculizumab monotherapy, and they have remained stable for about 12 months now. This report suggests that eculizumab has the potential to be used as a monotherapy, although a larger study with more extended periods of observation will be needed to further validate this conclusion. This will be the end of my presentation. I would like to thank Dr. Marabian for mentoring me on this project, and I'm happy to take any questions. Thank you.
Perfect. Thank you so much, Dr. Lee. That was a great talk and simulated some good discussion. I think the first question was prednisone considered? And then after that, Rick had some very nice discussion he brought up, which I would assume he'd like to kind of bring up. So maybe let's answer that first prednisone question, and then Rick, I think.
Question. Actually, interestingly, for these 2 patients, I think for patient number 1, we tried prednisone for about like 4 months in 2014, and then she had like worsening mood issues. She has like some underlying depression. So after starting the prednisone, she really had like more mood side effect, and she wasn't able to sleep and all that. So we had to like stop that. So we give that a try, but that wasn't very successful. And for the other patients, she actually have some like known allergy to the prednisone, like having rash, hypotension. So that's like a contraindication to try prednisone in her case.
Is it okay if I go, Melissa? So yeah, I think that was good points by Dr. Gonzalez-Perez. But you demonstrated you can manage these patients without prednisone, so that's pretty cool. So the issue is there really an issue of IVIG messing up eculizumab? I need to know that from the experts. You, Dr. Howard, I because I have people on both, but I think we need to know as a neuromuscular community, is that a real concern or not?
This Dr. Howard.
There he is. I chatted him. I said, Chip, come in the conversation. But Dr. Lee, you take a first shot at it.
Yeah, I mean, I don't have definitive evidence to say it for sure has interaction. Maybe Dr. Howards will have better answer. But I guess there's some concern about IVIG messing up with the recycling of the FcRn receptor and reducing the concentration of the eculizumab. But I don't know like 100% that this
That's
is
fair. That's fair.
Yeah.
Well, let's go to the source.
You're putting me on the spot, right? So the data suggests that IVIG activates complement, turns the system on. And in that manner, though there is no formal study looking at this head-to-head, increases the possibility that with increased complement activation, you have increased terminal complement complex or membrane attack complex being formed to attack the post-junctional membrane. That's the big problem.
Okay. But it is okay. But a lot of us are doing it, and we're seeing good effects from eculizumab. So, and I think the point is that we're able to reduce the IVIG. So, any other thoughts on that from other MSGers who are using a lot of eculizumab now? Guess not. I guess I'm the only one, Dr. Lee.
I saw there's some question about the use of azathioprine or rituximab.
Oh yeah.
I mean, we haven't tried that in our patient. I guess azathioprine, I guess both of these patients are pretty severe at onset and it takes a long time to work. So I guess, we're choosing something that, work relatively fast. So azathioprine wasn't a part of our consideration. So we really haven't tried that in both of those patients. And honestly, we haven't tried rituximab in those patients. We just go straight with the eculizumab.
Great. And then we had a question on, did this patient get a meningitis vaccine prior to the
Yes.
ECU? Okay.
Yes, of course. Yeah.
And then there's another one that was sent directly to me, but I can kind of read it to you. Any data that it changes the course of the disease in the long run?
I mean, right now we've been monitoring this patient for about 2 years. So I guess we still don't have enough data to predict the long-term course. I guess with time, we should be able to have more data and more patients to further, support this conclusion. Is it gonna, change the long-term outcome? But I guess this is a preliminary report on how well eculizumab is doing as a monotherapy, but I guess we'll have more data in the future as the time.
Yeah, I think, yeah, and that's your answer is totally right based on your two cases.
Mm-hmm.
I think what they're getting at is, do we have any bigger data internationally yet that eculizumab alters the course of MG, such as that some are going into complete remission or something like that? And
Yeah.
I assume we don't have that data.
No, I don't think we have anything like that right now.
There was a question for Chip. Chip, have you been able to withdraw some patients? I'm not positive who that was directed to, but that's kind of the last question we have in chat. And I think we.
Oh, David Johnson. Did you see that, Melissa? One of the MSLs from Alexion. He's saying there's some data saying there's some drug concentration reduction, but I'm sorry, that was based on my question. But anyway, it is in there. Oh, that's just to me directly. So this, I think, has to do with the IVIG. There's some data saying there's some drug concentration reduction if you give eculizumab with IVIG. So I don't know if that's published yet.
So to answer your question regarding, I've not been able to withdraw drug. And what's interesting, and we're putting the paper together now, is only in the rare instance where eculizumab or any complement inhibitor will normalize single fiber AMG, suggesting that there is a continuing neuromuscular block by the role of the antibody. And despite the fact these people are in minimal symptom expression, with ADL scores of 0, we have jitter values that are in excess of 100. Now, what does that say in terms of the correlation of jitter to clinical weakness? And that's fair. But to me, it represents that there is still ongoing blockade at the site of the receptor by the antibody, and would tell me that withdrawal of ECU or whatever, you're going to end up with a deterioration clinically. And while I've talked to many of you, I believe you need to attack the mechanisms at both ends to directly attack the antibody, but also to use complement inhibition and protect the house so it doesn't get destroyed, so to speak.
That's great, Chip. And you would have that data since that you're one of the few people that have that continuous single fiber data. So that's very helpful.
Great. That puts us at time for this session. Rick, I believe I hand it over to you.
It puts us at time for the session and the meeting. Sorry, I was muted. I was talking and I was muted. So end of session and basically end of meeting. That was a great way to end the meeting, Dr. Lee. I was actually surprised we didn't have more abstracts on eculizumab, since it's such a huge game changer in the management of MG. But maybe next year. So great meeting, right, Michael? What can I say?
Yeah, I think it's one of the best ever, actually. I think it was really, really good. I enjoyed all of today's sessions. I think the genetics session really highlights the challenge we have with variants of unknown significance. It's a real issue which I don't know whether the neuromuscular community need to think of better ways to handle that for our particular genes. But yeah, great talk, and I really enjoyed all of the meetings. So thank you to all the speakers and contributors, and obviously thank Liz and all the team for putting it on. So I think it was great. But I'm looking forward to a face-to-face meeting next year.
Yeah. Even Tony liked it. Look, you want to say something, Tony? You gave us a big shout out.
Oh, that's unusual.
No, I agree. That was one of the best meetings ever. Except for like when we met in Chicago, like the first time in the airport. How many years ago was that?
Over 20.
This is great. I'm looking forward to next year.
Yeah, I hope you'll be in Italy, Tony.
Before everybody goes, we're going to try to take a big picture.
Yeah, how do we do group pictures? How do we do that?